Hello, everybody. Welcome to Creation Myths. We're here tonight to talk about some really interesting stuff. We have a recently published book chapter from a number of... Oh, good. We are live. Good. Okay, good. I always just check myself, make sure we're good. Okay, so we are here tonight to talk about the book chapter that is... it's linked down below in the description. So if you haven't checked that out, there's a PDF link down there. Just pull it up. You have the PDF. That's the chapter we're going to go through. This is a book chapter in a long reference work. The chapter is called Dynamical Systems and Fitness Maximization in Evolutionary Biology. It is by William Basener, Salvador Cordova, Ola Hössjer, and John Sanford. Dapper Dino, can you correct my pronunciation on Dr. Hössjer? I don't.
I actually am not sure exactly what language that is, other than it's like kind of Scandinavian. So, other than saying that J should be a Y sound, so it should be something like
Hössjer. That makes sense.
Hössjer.
Yeah, that Hössjer.
Hössjer.
Okay.
Probably something close to Hössjer would probably be roughly accurate,
Thank
but
you.
Not knowing exactly which language, I can't really say. Sorry.
Okay, fair enough. So we have our four, and I'm gonna go ahead and say our four creation scientists, even though I don't think that's well established for at least one of them. But I know there are creation scientists. This is a piece of creation science. So before we get into this, we're gonna talk about this because this is a book chapter and a legitimate book. It's from Springer, which is the parent publisher for like the Nature publishing group. Like this is a legit scientific publication. So this is creation scientists publishing in the real scientific realm. It's not strictly speaking the peer-reviewed literature because it's a book chapter, not a paper, but it's, you know what, credit where it's due. So before we get into all the fun population genetics here, let's go around, introduce yourselves. Deadly Dakota Raptor, welcome. It's been a while.
Yeah, I'm Deadly Dakota Raptor. It's been a while. An engineer by trade. Just like to hobby around in the creationist sphere sometimes. Thank you for having me on.
Thank you. Dapper Dino.
I'm Dapper Dino. I also have a channel. It's called Dapper Dino. I'm the brown Dapper Dino, not the green one. There are two of them. Two of us, I should say. It focuses primarily on debunking creationism, especially young Earth. But there's also other stuff on there. There's some medical pseudoscience, some new age stuff. There's just some straight up, here's some cool stuff from science that you might not know. And I'm here. I consider myself friendly with Salvador Cordova, one of the authors here, the second listed author. And I'm basically going to be assuming that he's going to watch this. So sometimes I may direct my comments directly to Sal. So I'm just going to put that there right now because I'm pretty sure that there's a very good chance he's going to watch this.
Yeah, I want to second that. Also, I consider myself friends with Sal at this point, which is cool. So hi Sal, I hope you do watch this, and yeah, also we'll be talking about this at some point. So yeah, Atheist Junior, welcome.
Hey Dan, thanks for having me. Yeah, my channel name is Atheist Junior, and I also sort of make content debunking creationists, particularly Kent Hovind, and I did it before all of the news articles did it. I just want to say that I got it on the ground floor. So.
I want to direct everyone to Atheist Junior's channel because he somehow gets all kinds of fun stuff from Dinosaur Adventureland, and is just breaking news on Kent being awful left and right. So yeah,
far more awful than you think.
Far more what?
Far more awful than you think. Like just kind of amazing.
Just it is just awful all the way down.
Yeah.
So go subscribe to Atheist Junior for all your Kent Hovind news if you are not already.
Yeah, and I just wanted to say real quick that me and Paul Uji are collaborating on a big video about Kent Hovind that's going to be a timeline from just the past few months, and that's going to be coming out hopefully soon. So, okay, I'm done.
That's exciting. That's
yes,
very cool. All right, and David Neff, welcome back.
Yeah, thank you so much for having me on your channel. I really appreciate it. So my name is David. I am a biochemistry and molecular biology major at the University of Maryland, Baltimore County. I'm passionate about science education and evolutionary biology and viruses. So that's one thing Dr. Dan and I have in common. On my channel, we do a lot of interviews and discussions on science topics. Dr. Dan has been on my channel a couple times. As of recently, you should totally check those interviews out. We spoke about how to determine good science from bad science. And I also want to plug this in. This past Saturday, I actually had the privilege of meeting Salvador Cordova and Dr. Robert Carter because he went to a church near me and I just had to attend. So, Sal, I love you, man. I do really like Sal Cordova. He's a great guy. And it was a great pleasure meeting him. And I have lots of thoughts about the Dr. Carter event. And if you come back to Dr. Dan's channel on Monday, he and I will be talking about that, I believe.
Yes. Thank you for that. Thank you for that plug. So hi. If you don't know me, I don't know what you're doing here, but I'm Dr. Dan. The channel is Creation Myths. Previously just Dan, but apparently YouTube has taken to calling me Dr. Dan because I technically have earned that title. So, we're just going to roll with it. We're going to go with Dr. Dan now. And so what we do here on Creation Myths is we debunk really awful creation science and sometimes marginally less awful creation science. And we also have fun conversations with creation scientists like the aforementioned Sal Cordova and Dr. Michael Behe and some other people that have come in and out over the however long this has been going on. Yeah, so before we get into this chapter, I just want to note that I'm actually going to be chatting with Sal on this over at his channel, Evidence and Reasons. That's Evidence and Reason or Reasons. I always forget. But that's going to be
Reasons.
next Reasons. So that's going to be on Evidence and Reasons next Wednesday. That's not 2 days from now. That's 9 days from now. That's October the 13th, I think. Sal and I are actually going to talk about this book chapter, so I'm really excited for that conversation to actually have a conversation with one of the authors. So that all being said, thank you all for being here. Thank you for the 17 people who are here to watch us talk about population genetics for however long it takes. Great, thank you. That makes me so happy. So let's just go around and we'll start with David. We did you last for the introduction, so let's start with you. Let's just get kind of big picture takeaways from this in a minute or two, and then once we get kind of everyone's top level thoughts, we can kind of work our way through the specifics and really go into it.
Yeah, sure. So the first thing I noticed while I was going through it is a very dishonest quote mine in the conclusion, and we're going to get to that. They pretty much go through the creationist bingo card: genetic entropy, drink; Muller's ratchet, drink; the waiting time problem, drink. So it's tough that we've already discussed many times before. Yeah. Don't actually do the drinking game because you'd be dead. Instead, just do the bingo card instead. So yeah, those are my initial thoughts.
Yeah, Atheist Junior, what do you have? Oh, that's very very fancy background.
Well, I figured it was appropriate. Something that jumped out to me was just this part that's talking about the eclipse of Darwinism, and I feel like this is something that like creationists do a lot. Like instead of calling it evolution, they're going to call it Darwinism because they want to lump in a lot of other things or sort of conflate social Darwinism with evolution. And it's just talking about like how Darwin, like, oh, well, he didn't know about Mendelian genetics and his theory at the time was incomplete. And it's like, well, yeah, I mean, what's your point? Like, I don't know. It's true, but
and in that same section of the paper, as we go through, people might notice that they talk about the eclipse of Darwinism and go through the history of the development of like Mendelian genetics and population genetics in the early 20th century. But oddly enough, that section kind of stops at like 1950, and it doesn't.
Weird.
Like there's more.
Yeah.
There's more.
Weird.
And so when we get to that section, you'll
up.
see it. Yeah, it just like and then the 1950s, and then that's the end. I'm like, well, but they're okay. Yeah. Dapper, what did you think of all of this?
So I actually am just fresh off of taking a look at Sal's presentation of this, which actually, interestingly enough, didn't seem to dive too deeply into the I guess chapter. I want to say paper, but I guess it's a chapter. I don't know. I'm probably just going to use...
Yeah. So here, I'm actually gonna pull up the cover of the book just so everyone can see it while you're talking, because it is a book chapter, not a paper.
There's a fair chance that I'm going to say paper, and I mean chapter, and I know that, but it's just the word paper is so just stuck in my brain talking about this kind of thing that I apologize in advance. I can almost guarantee it'll happen. But anyway, dive too deeply into the text of the chapter itself. What they did do is what's Precious Sal's own take on this.
Yeah, this is your...
Is that a lot of it was talking about why it is that they think that they should be allowed to change the definition of fitness. And this is directed at Sal. Sal, look, I get that you don't like the definition of fitness that science uses. I get that. I'm not saying that you shouldn't dislike it. But what I'm saying is, when scientists come up with a model based on definitions and they're not seeing a problem, you cannot come in and say, if you don't find it the way you do, then I can invent a problem and be taken seriously. You can't do that. If you want to find a problem with the model as it is, that's great. You're going to have to work within the model to find the problem. You can't just say, oh, I think that intelligence is just an inherent fitness advantage by how I just arbitrarily define fitness. Therefore, anything that loses intelligence over time is a decrease in fitness. Therefore, genetic entropy. You just can't do it, man. You have to show some actual problem. And another thing was there was this big assumption that increases in complexity is a problem for evolutionary biology, and that evolutionary biologists are trying to solve that problem. That has never been an acknowledged problem. If you want to say that that problem exists, you first have to show it. You have to actually find the problem, and you can't start by redefining the terms of evolutionary biology to pretend a problem exists that doesn't. And that's my biggest takeaway on what is freshest in my mind, which is Sal's presentation of this on his own channel.
So I do want to say, I want to stick a pin in that redefinition, because that is the linchpin of the whole thing. Because the whole thing, and this is going to be my big takeaway in a minute, the whole thing is based on the whole argument that's constructed is based on a bait and switch that hinges on that redefinition. We'll get to it.
Can I mention something?
Oh,
sorry.
please.
Yeah, I just wanted to mention a line that I forgot. I'll be quick. And it's the same section, I think, and it says Darwin's theory lacked traction in the biological community beyond its initial interests. And I'm just thinking, like, are you kidding me? That's just wrong. Like, and they also say biologists saw Darwinism as incompatible with Mendelian genetics, but I don't think that's true either. I feel like you have Mendelian genetics stacked upon it, and then other things, and that's sort of what we call like the modern synthesis. So,
right, and to be clear, there is no such thing as Darwinism, Darwinist, and there's no such thing as an evolutionist either.
Yeah. You can use Darwinism correctly to like refer to the ideas presented by Darwin in 1859, 1860s, right into the 1870s. That's what Darwinism is. That isn't a thing anymore in evolutionary biology. We've moved on. All right, Deadly Dakota Raptor, what's your big takeaway here?
Well, not as in depth on some of the specific genetics, but it just feels like there's so much putting the cart before the horse, because they spend so much time on their math and derivations trying to rehabilitate all the Fisher's formulas. They recognize all the problems that have with that, and just try to like reincorporate and back fix it in. It's just, to me, it screams like they're trying to update the Fisher's problem to fix what they want it to present, to try to at least do all this work to hide the fact that they're trying not to address it.
Yeah, it's so the Fisher, the kind of the updated version of Fisher actually goes back to a 2018 paper by Basener and Sanford, who are two of the authors here. They did a 2018 paper on Fisher's fundamental theorem and the whole thing. And actually some of the figures in this chapter are the same. I don't know if they're exactly like if they're directly taken, but they're the same basic figures as from that paper. So my big, we have one more guest. This is delightful. Hello. Welcome, Speed of Sound. It's so nice to have you. I'm so glad you joined us. Please tell us, introduce yourself.
I'm Speed of Sound from Answers in Atheism, and I have not read through the entire math section of the paper, just read the beginning of it. And I have, but I've been working with Cell for a while on this fitness problem, and I have a few comments. If
Yeah,
one
what's
of them,
what's your big takeaway?
okay, in the beginning, it seems like they were trying to poke holes or shovel a little bit of dirt on the whole idea of Fisher's fundamental theorem, which a lot of people don't really run into so much in population genetics. And the other thing that they're trying to do is come down on fitness. And it's interesting. He refers to Lewontin's 2003 paper, I think, on fitness, where Lewontin had a lot of problems with fitness as a scalar number. And Lewontin was pointing out that when you look at like actual sexually reproducing populations that have overlaps and have finite population sizes, that it's very difficult to come up with a with a number basically for fitness, that it's more like an equation or like a differential equation.
Right. Yeah,
and
because
then
when you don't have discrete generations, when it's just a rolling wave of reproduction, then the simplified way of viewing it kind of breaks down. Right.
So that was primarily what Lewontin says, and then of course the Cell and his ilk are taking it up, and they're trying to bring in some kind of concept. It's almost like they are confused themselves, yet they criticize this idea of fitness as the best and the most perfect, right? And they talk about engineering. And then they double down and they want to make fitness about perfect engineering. And they keep saying things like, well, it's obvious that if a cavefish loses its eyes, that it's not more highly engineered, that it's, just lost some ability, right? And it might increase its fitness. But the thing is that fitness was never about this perfect engineering idea. And like I pointed out to him, it could be that the human race will get dumber and dumber and actually survive better and better because of it, right? We don't know. In any population, you can't define fitness by engineering principles or engineering perfection the way he wants to. And I think they want to turn it, but they want to turn it into that so that they can prove that with many populations that wouldn't happen, right? That you wouldn't end up with a finer and finer tune. Even though when you look at molecular mechanisms, you see what looks like a hell of a lot of fine-tuned engineering, right? Because it manages to, over many generations, select out a very, very fine, clean pathway that works, and it works very well. So I don't know. And then I'm guessing that what they did with their mathematics is after they wrote upon the criticism of models for population genetics, that they went ahead and increased a more complicated model to show them the results that they wanted.
Can you guess which one?
No. Which one?
Mendel's accountant.
Oh, so because I spotted that later on, and that was when I quit reading.
You mean the one whose predictions have been routinely falsified in actual organisms?
The one that you can't make match actual observations no matter how hard you try. You literally can't get Mendel's accountant to match the outcomes of the Lenski experiment.
And it's horribly undocumented code. It is undocumented code.
This is for Sal, who I think is watching.
Here, before we get, I want to hear Dapper, and then that way, Dapper, you're over there. And then before we get into that, I want to give my 30,000 foot view, and then we can go chapter and verse.
Sal, until you can get Mendel's accountant to match real world results, it is useless. You might as well reference like a 5-year-old's crayon drawing of a bacterium, because it's exactly as useful. It's much more sophisticated because it can do lots of fancy math, but it doesn't any more match reality than a 5-year-old's crayon drawing of a bacterium.
Right.
So on that, I just want to say there's a saying that many of you probably know. If you haven't heard this, you should know this saying. It's a very useful saying. All models are wrong. Some are useful. Because all models, and they talk about this in the chapter, that all models are necessarily a simplification of a more complex system, so they're never going to be exactly right. But they can be useful in that they help you learn something about whatever system it is that you're modeling. And some models do not do that. And I put Mendel's account in the latter category because it is it does not make it easy to set realistic parameters that then match experimental results in Mendel's account. That's not a thing that you could readily do, which is a problem. So I just want to take a couple minutes here and give my big picture look at this chapter. So for anyone who has not looked at the PDF, I recommend you do it just to see it. It's long. It's 60 something pages. It's like 60. The conclusion is it ends on page 67, but it starts on page 2. So
and we're going to go through every single word.
No, we're not. We'll be here all night. But here's the big thing that I want people to take away from this. If you get nothing else out of this, if you just skim it, if you don't look at it, I encourage you. I think you should read it, even if you're not a biologist. I think you should read it if you're interested in this conversation. But here's the argument they make. It has 3 parts. Part 1 is that this idea of universal fitness maximization is unrealistic in biological systems. That's basically the first argument they make. And this is where they're talking all about Fisher's fundamental theorem and how it's not really valid and you need to include other factors in it. That's basically correct. Speed alluded to this a few minutes ago that like in modern evolutionary biology, Fisher's fundamental theorem like doesn't come up all that much. I know this because I didn't ever hear about it in grad school once. And like I studied evolutionary biology. I'm an evolutionary biologist and it never came up in grad school. It's just not like a thing because it's so oversimplified. There's better ways to examine things like how fitness changes over time than using Fisher's theorem, which is from the early 20th century. We've learned a lot about how this stuff works since then. So the first argument in this paper is basically that Fisher's fundamental theorem is flawed and not realistic, which is to say that you shouldn't expect universally fitness to just maximize over time. And that's true. That's a valid thing to argue. Perfectly fair. The second part of this argument is that fitness should not mean reproductive success. Fitness should instead mean functional complexity in some way, the total functional what's going on within a biological system, within an
which
organism,
is poorly defined.
Which is extremely poorly defined and has a ton of problems with it. Because the correct definition of fitness in its simplest form is reproductive success relative to other members of your population. Basically, how do you do compared to everyone else? The more mathematical way to look at it is the relative frequency of your alleles in the next generation compared to the alleles of other individuals in the population, right? How do you do compared to them? Do they get more frequent? Do they get less frequent? Are they average, right? That's what we mean when we talk about fitness. Now, that is useful because everything in evolution is context dependent, right? What works in one environment is not going to work in another environment. Those blind cave fish are not going to do well in the open ocean. Eyes are really useful in that situation, right? So something that's high fitness over here is going to be low fitness over here and vice versa. And if you treat it as, once you come up with a way of quantifying, functional complexity or total functionality of a genome or whatever, once you come up with a way to quantify that, that's an absolute measure. It's no longer context specific. So it doesn't tell you anything about how well something is going to compete and whether its traits are likely or unlikely to persist within a population, right? So that's the first problem with the redefinition. And that's just putting aside you shouldn't just redefine terms in the field that you're criticizing, as Dapper said earlier. Like, it doesn't work for what fitness does in evolution.
Yeah.
Redefining it now doesn't do that thing, right? So putting aside that you're taking a well-established term with a well-established definition and changing it, and insisting that everyone use the new definition doesn't work for what the term does.
Right.
So
I think
it doesn't work.
It's important to note that the reason we talk fitness the way we do is because what we're concerned with is what will the population look like in the future? Yeah. And the only way to answer that question is to say, well, which alleles are increasing in frequency and which ones are decreasing? Anything else?
And natural selection is going to drive that, at least in part.
Right. And any other way that you measure that isn't going to answer that question. It'll answer a different question maybe, which is fine if you're asking a different question. But that's not the question of evolutionary biology. The question of evolutionary biology tends to boil down in various ways to what will this population look like at some point in the future where we can still predict it? Because, I mean, you know, you go like a trillion years, who knows? Right, and why? Yeah. And that's why fitness has the definition it does, because if the allele frequency of this allele is going up, well, guess what? We know in at least some amount of time what the population is going to look like down the road. It's going to have more of that allele.
We can make predictions. Yeah. So that's the second thing. Is the second part of this argument is redefining fitness. The third part is the key thing. The third part is they're saying, okay, this fitness maximization doesn't work. Here's Fisher's math. Here's our math. Their math doesn't work. You don't get maximization. Okay. Second part, also, this term fitness, this should mean something else. Instead of reproductive success, it should mean functional complexity, right? Total functionality. Part 3 of the argument, because we can't maximize fitness, and fitness, according to us, the authors, now means functional complexity, there is a fundamental problem with evolutionary theory because we just showed mathematically that you can't maximize complexity over time. That's the bait and switch. They're taking the math that's using the one definition of fitness, substituting a different definition of fitness, and then saying, therefore, biological systems can't get, we don't know, like the field, evolution, does not have a mechanism or an explanation for how systems get more complex over time. We just showed mathematically that they actually can't/don't. That argument only works because of the redefinition of fitness in step 2. But all the math that they're using and all the math that they're criticizing is based on the correct definition of fitness, not their redefinition of fitness. So the entire argument, it's like I said, there's 60 something pages of argument in here with a lot of math, but it's all a bait and switch where they're using math and showing one thing happens with that math. But that math is based on one definition of fitness, the correct definition of fitness. Then they turn around and argue, no, we should use this other definition of fitness. But the math still works. We're going to use the same math, but we're going to change the definition. Therefore, you can't maximize fitness. Fitness is complexity. Complex things can't evolve, right? That's the argument, and that is not going to fly.
The one thing I would say there is I'm a little uncomfortable saying it's an incorrect definition of fitness, because the thing is, you can have a different conversation with that definition of fitness. It's just not evolutionary biology, right? Like saying total functionality as a thing that you want to talk about is fine, right? Like we can have that conversation. It's just not the conversation that evolutionary biologists are having when they talk about evolutionary biology. So it's not so much that it's intrinsically a bad definition. You're just using it to replace a definition that's already operating, that we're already working on, and it's already answering a certain subset of questions that you can ask about organisms.
Yeah, people are already building models.
Right. If you want to talk about how biologically complex this particular paramecium is, or how great the acuity of an eagle eye is, and you want to talk about how fit they are in those terms, it's a little weird to use the word fitness, but it's not a bad conversation to have. Those are cool things. We should talk about them. But you can't then pretend that your definition of fitness should override the definition of fitness in evolutionary biology. So it's less that it's wrong. It's just that it's incorrect to import your idea from a different field into this one and pretend this is the one the field should be using.
Yeah.
The other thing about models and what they do is that we'll use those fitness numbers, relative fitness, perhaps on a gene, to give us an idea of how a system would behave ideally. Proving that the model doesn't work or the model has some finding is not necessarily proving what is going to happen to an actual population. What it does is it tells us what direction the model's going if we fix all these parameters and we let these parameters vary, right? I'm working on a very complicated model right now, and we're getting, and I wrote the model, but we are getting surprising results that are surprising us because we put the best parameters into the model that we can find. We put the best machinery into the model that we can find, and we're learning something about what will happen if this particular scenario played out. But it doesn't have a thing to do with what would actually happen.
Right.
Well, it does. It tells us which way the wind is blowing, or if the wind blows this fast and, this hard and this long, this will happen, right? That's what we can tell with models. So I think they're kind of conflating. They think that they're disproving evolution with a model. And that is no more valid than trying to prove evolution with a model, because I don't think we do that.
So I want to, I'm happy. I mean, it's already 9:30 and I don't know how long I'm going to do this, but I'm happy to go top to bottom if we just want to start with the abstract and go from there and see if anybody has any thoughts they want to do. And I'm going to pull up the paper, which is, I vomited highlighting and comments all over mine. So I'm going to put this up. I don't know what the review I want to say right now, I don't know what the process of review was for this. I don't know. There's a good question and I don't know the answer.
Dr. Dan, I hate to do this, but I just stopped in to say my piece, basically.
Well, thank you for coming.
And I'm going to go watch and eat dinner.
All right, cool. Enjoy. Have a good dinner. Good seeing you.
Okay, I might pop in again if I'm so compelled. We'll see where it goes, but I will be watching. Okay.
Awesome.
Good enough. Later.
Is it okay if I make a comment real quick?
Please make a comment.
So I think you guys were saying that they're redefining terms, but I think another thing that they're doing is sort of like fabricating terms out of whole cloth. Like there's one section about reductive evolution, which I don't think is a thing. I've never heard of that, but basically it says reductive evolution involves the loss of genetic information and the loss of biological functions in a population over time. And then it says, if we define fitness realistically, reductive evolution is really evolution going backwards. And then they quote Haldane saying most mutations lead to a loss of complexity. And I feel like this paper it's trying to come off as, like really somewhat scientific, but really this is a Kent Hovind level argument, because it's funny that you mentioned the cavefish earlier, because one of the arguments he makes is that, like cavefish losing their eyesight, it's the opposite of evolution, because it's not gaining something, it's losing something. And I think we're all familiar with the creationist tier list, and Kent Hovind is on the bottom, but this argument is trying to punch above its weight, I think.
I think that's fair. I think that's fair. Yeah, there are some arguments in here that are really sophisticated. But then they get to this reductive evolution part, and you're right, because it's talking about like evolution going backwards, and that's just not a thing in evolution, right? Like stuff changes, and we don't talk about forwards evolution and backwards evolution. It's just evolution,
right?
And framing it that way implies like a goal or an ideal, and that's imposing a value judgment on top of these processes that are happening, completely independent of any of that type of framework.
The only thing I can even imagine as being somewhat easily described as quote unquote backwards evolution would be like, let's say you have like a pseudogene, right? And you happen to have a mutation that manages to fix it, which would be extremely unlikely, but I can't say possible. And then, okay, now you have recovered an ancestral trait as what starts out as and maybe it spreads to the population. That's the only case I could ever see where like, maybe that could be reasonably described as backwards evolution. But going from your idea of complex to your idea of simple, which again, complexity not well defined.
I want to make sure we get David and Dakota in here. So if either of you two would like to chime in, please.
Yeah, I do have to get going pretty soon. I'm starting to feel really tired. But if you do not mind, I would like to jump down towards the end where I noticed a big dishonest quote mine from Charles Darwin.
Oh yeah, let's just jump. Yeah, because this is a great ending. Come on.
Yeah, because I got to get down soon.
Here, we'll jump all the way down here. It's on page 67. I know exactly what you're going for. As you can see, I got a lot of thoughts about this chapter. It's a lot of highlighting. Where are we? We're almost there. 59, 61. If you're really sharp-eyed, then you notice there was a graph from Lenski in there.
So much highlighting.
So many thoughts. Okay, here you go. There you go.
Yeah, sure. So, yeah, sure. So notice how they deliberately misquote Darwin. In Darwin's day, there was no such thing as biophysics. Even Darwin saw the problem. Darwin cited the organs of a extreme protection and complication. And said, to my suppose the eye, with all its animal, contrivance, ... Here's a pro tip for everyone. If you're reading a book or a paper and there's a long quote and there's a bunch of eclipses, there's probably a reason that those parts are in eclipses. So you might want to go and go to the original place and see what the original quote actually says. And yes, I do. Sorry, I do. Organs. My mind is all mixed up. People are making fun of me in the private chats because I cannot read to save my life. I am very dyslexic.
You're
So
good.
yeah, I'm very dyslexic. So sometimes I will read something that I know what it says and then read it completely wrong.
To you, no one here thinks less of you.
Yeah. Yeah, that's right. If you ever see me reading a PowerPoint and you see me tripping up on words, that's why. Because I'm sure you guys noticed that before in my presentations and stuff.
You're good.
Yeah,
You're
sure.
good. Don't worry.
Yeah. So
So
here's what Darwin actually said. Once I try, my computer is going very slow.
Here we go.
Yeah. To suppose that the eye, with all its inimitable contrivance for adjusting the focus to different instances, I freely confess absurd in the highest degree. Yet reason tells me that if numerous gradations from a perfect and complex eye to one very imperfect and simple, each grade being useful to its processor, can be shown to exist, if further the eye does vary ever so slightly, and the variation to be inherited, which is certainly the case, and if any variation or modification in the organ be ever useful to an animal under changing conditions, then the difficulty of believing that a perfect and complex eye could be formed by natural selection, though insuperable by our imagination, can hardly be considered real. How a nerve comes to be sensitive to light hardly concerns us more than how life itself first originated. But I may remark that several facts that make me suspect that any sensitive nerve may be rendered sensitive to light, and likewise to those forcers vibrations in the air which produce sound. So that is what Darwin said. And notice how they cut all of that out and deliberately put mind to make it say that Darwin said that the evolution in the eye is absurd. No,
Yeah.
And that there can't be any possible explanation. Yet we have shown experimentally, and we have shown how light and eyesight can evolve. This is one of the most this is probably one of the most quote mind statements in all of evolutionary biology. And so, yeah. Pro tip: if there's an eclipse, go back to the original quote and read what's in the eclipse, because there's probably something important that the author decided to skip. So that's my big takeaway.
Yeah, you can see my thoughts right here. You can see my thoughts right here. Just like do better than this. Whoever wrote this, I don't know which author is responsible for putting this in here, but like whichever of the four of you, do better than this.
Ultimately, they're all responsible because they all signed off on it.
Yeah, this is a classic quote line.
It's true.
Classic.
By the way, quick little thing. I do want to. I do see what's going on in live chat here, and I do want to say, I've exchanged words with Sal over the years. Some of them polite, some of them less so. But Sal and I, we consider ourselves friends now. And I don't want to make this about Sal in the chat. So just lay off, okay? We're gonna talk about the paper. We're gonna talk about what's in the paper. We're gonna talk about whether it makes sense. A lot of it doesn't. But we're not gonna go back and dig up blog posts from the 2000 aughts because people fighting on the internet. We're not going to do that. That's not
Yeah.
relevant to what we're talking about. So just
Right.
keep that in mind, everybody.
Yep. So,
Yeah.
Dr. Dan, I gotta get going because I'm exhausted. Thank you so much for having me on.
Oh,
I really appreciate it. And join us on Monday because we are going to be having fun talking about the Bachelor Quarter event that I went to on
I'm so
Saturday.
excited. That'll be next
Yes.
Monday with David Neff. I'm pumped. Thank you, David,
Yes.
and we'll see you soon.
Yeah, and Sal, I love you, man. If you're watching, it was great meeting you last Saturday. I really hope to see you again in person.
So yeah, I just want to co-sign what David was saying there is that this is one of the most commonly misquoted statements in all of like the evolution creation discussion that goes on, because creationists take this and they say, hey, Darwin said, look, how could this have happened? He acknowledges the problem, but Darwin is setting up the question, and then in the rest of the paragraph, he answers the question.
Yeah,
You can't just cut out the part.
It's the opposite of what they're trying to say it is.
Yeah.
You're trying to say this is Darwin admitting a problem. It's Darwin showing why it isn't a problem.
Yeah, I mean, he goes on for most of the chapter really talking about this.
And the thing is,
Yeah.
what is this actually? This is cited, right? Yeah, Darwin. Well, they cited it incorrectly because there is no such publication as Darwin 1959.
Yeah, good catch. Yeah, it should be 1850. It's origin
1850.
species is what they're. Yeah, it's origin
Right.
species.
So the thing is, okay, if you cite something, you should have read it, and not just the tiny bit that you're pulling out. You should have read at least a few pages maybe before and after.
Dapper, would it surprise you that problem comes up several times in this chapter?
No, because it's creationism.
It comes up several times.
So
it comes up several times.
I don't know who checked this and everything, but it is a common problem with creationists that rather than actually saying they cited a work, when actually what they've cited is a creationist collection of quotes, which is literally just an index of quote mines, because those are all over the place. It's very easy to find lists of quotes that creationists can use to make their point as just a laundry list of dishonesty. But the thing is,
go ahead.
They don't bother to check back to the original. So the creationist in question who's quoting it probably actually hasn't read the original, and so they're only dishonest in the sense that they didn't really check, but they're not. I don't think they're actually lying about what they think that this thing says. Like, I bet that all the authors in this actually think that Darwin was admitting a problem. I think that they all think that. They're wrong about it. And I hope that now that at least one of them is almost certainly hearing me, sorry about that, you can point out to your peers that no, you actually cannot use this quote this way. It is a red flag of intellectual dishonesty on at least some level.
Is it possible that they were citing the 100-year reprint of Origin, maybe?
Possibly, yeah.
Theoretically possible. Okay, let's go with that. Yeah. Atheist Junior, before you jump in here, can I just point out that this is another example of this right here? This quote right here that they pull from this Birch 2015 review. This paper, or 2016, it's the same paper. I don't now know if it's 2015 or 2016, but it's the same paper. This is from the abstract. The paper, where what they're saying here is basically people still treat fitness maximization as like a thing, right? And they quote this paper, right? He says this gets bad press in population genetics. Other areas are probably, attempting to maximize. Okay, so a couple things that I want to small things I want to point out here. One, as if attempting to maximize is not the same thing as actually maximizing it. And that's an important distinction because Fisher's theorem, which is what they're arguing against here, is about how populations actually do maximize it in practice. And we know that that's not correct for a lot of reasons. But that's the idea. So attempting, we're personifying it a little bit, but attempting to maximize it is not the same thing as actually doing so. Second thing, this paper is all about how it's context dependent. And we're saying, okay, in this specific context, in these specific situations, you can see maximization occurring, even if it's not a global maximum. Maybe it's a local maximum, right? It's a context-dependent maximum based on not just selection, but like all this other stuff going on, like mutations and drift and whatnot. So this, if you read this paper, which I did, it actually doesn't support what they're claiming it does here. This is not a proper citation, and that's not the only instance of this in this chapter. There's several others that I caught. So that's not great. And this is all the way up at the top. This is like right in the introduction. So the framing of the problem is like, this is a thing in modern evolutionary biology. The paper that they're citing is actually not saying that. So that's, not super great. Anyone have any thoughts on the introduction? Do I have more thoughts on the introduction? Let's see. I'll go through. Stop me if you see anything you want. Basic perspective is whether fundamental laws exist. The way they do in physics has evolved over 150 years. Okay, sure.
Well, I was going to say, if you could you go to page 50? That's the thing that I'm looking at. It's where it says, "Why are we not dead 100 times over?"
Oh, so we're gonna get to that in a little bit. I want to go through. I want to construct it in order. I don't want to jump all the way down there, but that section is important. That section is important because that phrase is it has a history. So we'll get there.
Okay.
So I'm just gonna scroll through mine, and if you want to stop anywhere, everyone just jump in. I don't have much to say about like the history of like Mendelian genetics and everything. Like I didn't find whatever.
I think what we're really gonna have something to talk about is when they stop talking about the history of evolutionary biology, because that's an interesting aspect of this.
So we're kind of getting there here, where we get to the eclipse of Darwinism, and then we get into population genetics, and now here's where we get into like Fisher and fitness maximization. This is just me pointing out important parts, like how he has his theory. That's fine, or his theorem, and that's great. We have some definitions, fitness landscapes. Going through all these definitions is beyond the scope of what we're doing here. But if anybody is unclear about any of the definitions, just ask in the chat, and I can try to just jump in if anyone's unclear about any of the definitions. But basically, we're talking about visualizing differences in fitness in a population. So they go through how basically people over the 20th century have realized that Fisher's model is overly simplistic, which is true. It's kind of akin to, if anyone's familiar with Hardy-Weinberg equilibrium and that concept of modeling an idealized population that's not evolving. It's a population that's not experiencing any natural selection or genetic drift, no non-random mating. So mating has to be completely random, has to have no mutations, right? So like the conditions for that are not realistic. They're never met, but it's a useful null hypothesis. And in the same regard, Fisher's fundamental theorem is kind of useful in the same way of like, okay, under a set of unrealistic conditions, here's what we expect to happen. That doesn't happen. Why not? Right. And now we can like learn from it. Right. And that's kind of the utility of it. It's not that it's literally supposed to be true. We have a super chat here that I'll pull up for a second. Thank you. Carrie Bronson for $5. When computation was done with a pen and paper, I had failed an essay if that was conversation. Just say, yeah, I agree. It is in fact, it's a chapter of a book, so it's not like a paper, strictly speaking. Yeah. So I think the conventions are a little bit different. But yeah, it is the language kind of. Yeah, I would love to see the breakdown of the authors of like who primarily wrote each section. I think that would be really interesting.
Look, my baseline for young Earth creationism though is like the raw Matt standing for truth book, and I mean compared to that, this is barely. Like, genius level like writing. Like look,
well, like we said, these are professionals,
right?
The fact that it's not all they've all published like real science, like give them that.
Yeah. So okay. It's whatever.
So this is where we get to like, how is Fisher's model unrealistic, right? It discounts linkage, recombination, and epistasis. Great, fine. that means it's not going to realistically model things. Absolutely, I agree. This is at the beginning, I said there were 3 parts to this argument. This is part 1. Fisher's unrealistic. Yes, it is. There's another way it's unrealistic, where down here, assuming that the phenotypic fitness, which is to say reproduction rate, is equal to like whatever you would expect based on the alleles, the genetic fitness, and we know that that's not true, right? There's phenotypic plasticity, there's genetic drift, there's all sorts of other things that are going to change that, right? So like we know that that's not the case.
It's almost like biology is complicated, and trying to reduce it to 3 terms is unrealistic.
Yeah, almost like that, right?
Yeah.
That's and
it's also almost like everyone already knows that.
Yeah, so this is a whole bunch of math, and like honestly, I there's a lot of the math that I'm cool with, and there's a lot of the math that I don't understand. So I didn't comment on the math because I'm commenting kind of upstream of the math on like the evolutionary theory of like are the underlying structures here on which they're basing their argument, is that a valid foundation? And the answer is no. So like the math is kind of almost besides the point to me, because the structure of the argument itself is not valid. And I mean that not in the strict logical sense.
I think you mean sound.
Yes, if we're being technical, thank you. Yes, it is not a sound argument.
Right. Because the thing is, you can have perfect math, but if you start out with unrealistic assumptions or definitions that don't work, well, then I'm glad that your math works out, but it doesn't mean anything in the real world.
Okay. Yeah. So here's something worth commenting on. Oh, Atheist Junior, go ahead.
Okay. I was I forget what video I was watching yesterday, but they were talking about some sort of like science study where they had a 99% rate that something was correct, but they explained that it's only 99% true if all of the assumptions that went into that are correct. So if those aren't right, then it's not really that high of a chance of it happening.
Yeah, exactly. And when we get down to the part that you pointed out, which is kind of the stealth genetic entropy section of the chapter, they'll talk about, they'll kind of like on the slide talk about like the assumptions of that model, that's John Sanford's genetic entropy hypothesis. The assumptions for that are just bonkers, like a constant distribution of fitness effects, like that doesn't change. That's not real.
Yeah.
That doesn't have,
or the idea that over time the proportion of mutations likely to be deleterious is going to stay the same. Like no.
It's just it's just the math doesn't work the way Sanford says. So like you could put together a model that like if all these impossible things happen, then you get his outcome. But like it's a series of impossible things that have to be the case in order for that to happen.
Every time the garden gnomes show up in my garden, the tree gets 4 feet taller. The only problem with this model is that the garden gnomes have yet to show up, so my tree stays at the normal tree growth rate.
Right.
Yeah, because there's an assumption that he's making going into that, and that he said something about like it's only if these deleterious mutations are like small enough that natural selection won't notice them, which I'm like, I don't know.
Which is a contradiction in terms. Yeah. It's because you define deleterious based on effect on reproductive output. If it is effectively neutral, if it is invisible to selection based on the population parameters, right, the size of the population, such the strength of selection, then it is effectively neutral, right? And it will not have a measurable effect on reproductive output, even if in a perfect world, if you had an infinitely sized population and perfectly sensitive selection, it might have a negative effect. In the real world, it doesn't. Therefore, it's not deleterious in practice, right? And it's a contradiction to say that these very slightly deleterious mutations will accumulate and cause the population to go extinct, but are also immune to selection.
And that's
those
still
two
the
things
case.
Can't happen.
Even if you emotionally dislike this allele, right? Because that's actually one of the things I pointed out to Sal when we had it was technically a debate, but it was more of a discussion about his definition of fitness, which is the one we're operating in here. And I call it a discussion because like there wasn't a whole lot of like attacking, and like it didn't have the same feel that debates tend to. It was a very nice conversation. But anyway, I pointed out you taking your human emotional connections to things like physical strength or visual acuity or intelligence that there's no reason why your human emotional evaluation of these things should matter to science. Science, to the greatest degree it can, tries to divorce itself from human emotions to say, okay, regardless of what I would prefer, what is actually happening? And all of these definitions that Sal and company use ultimately tie back to, well, it makes me feel warm and fuzzy if animals are more like this, and it makes me feel sad if they're more like that. Therefore, this is fit and that is unfit. And no, that's just not how anything works.
Yeah.
Sorry.
So we have two not new but significant errors right here that I've highlighted. First is a classic right here that we've seen, is the citing Kimura 1979 for the beneficial mutations are so rare as to be statistically undetectable. Now, you have other more recent papers on there, but the figure that he actually shows for this a little bit further down is adapted from this 1979 paper.
Mhm.
And raise your hand if you've heard this one before, that Kimura himself acknowledged that by the way, famous population geneticist Moto Kimura basically kind of, I don't want to say came up with the field of neutral theory, but his monograph, the neutral theory of molecular evolution, was like groundbreaking for
By
the
Dakota.
field by Dakota. I'm sorry. Was groundbreaking for the field of evolutionary biology. And he published this paper in 1979 about his model, and he said, we're not going to count beneficial mutations. Why did he do that? Dapper, I think we've talked about this.
We've talked about this a whole bunch. And interestingly enough,
Yeah.
I actually found out about this independently of you, which is one of the few times that like genetics and evolutionary biology have crossed my path, and it did involve you. But like, yeah, I was presented with this idea by creationists way back before I even knew you. And I looked up Kimura in 1979. I was like, oh, look, it says not that he's ignoring them because they're so rare and so unheard of. It's that if he puts in any level of beneficial mutations at all, they swamp the results, and all you get is selection for beneficial mutations. So in order to actually weed out the beneficial mutations and just look at neutral, he actually has to completely pretend that they don't exist at all, because any positive number of beneficial mutations ruins the signal of neutral variation, which is what he was specifically looking at. It's the exact opposite of what the creationists want it to be.
And I want to point out that I believe this section comes from John Sanford. He's written about this extensively before. It's in his book, Genetic Entropy and the Mystery of the Genome. He's been corrected on this specifically. Like, it has been pointed out to him that no, Kimura actually said they exist. And he actually in that paper says, here's the role that beneficial mutations play in this. I'm just not modeling them. Like, he actually explains exactly what he's doing in that paper. But creationists ignore that. And Sanford has actually been corrected in writing. And his response was, well, no, Kimura would see this the way I see it. Like, no.
He said otherwise.
He actually said the exact opposite of you. So this is just like, this is not honest. This part is not honest. I'm comfortable saying that this is not just wrong, it is dishonest.
Yeah, it's hard to imagine a way that you could cite Kimura that way, having read him, and think that this is what he meant.
So then we get to this part. Oh, go ahead if you have another.
No, it was just going to be a repeat of things I've already said. I don't need to do that, so let's keep going.
So then we get to this part right here, which is a really interesting confusion of two opposite things in population genetics. So bear with me a little bit. I'm going to nerd out for a second here.
Yay.
So what they're describing here, right? They're talking about mutation rates. They're talking about Muller's ratchet, mutation accumulation. But what they go into, this is the important part right here. Small populations, mutations can accumulate and drive fitness down, even causing extinction. Now, this is a thing called mutational meltdown, right here, or an extinction vortex. And the reason it's called an extinction vortex is it's, circling the drain, right? As a population goes extinct, it's a positive feedback loop of death, basically. So what happens in a small population is you have genetic drift predominating over selection, and you have an inbreeding. So what happens is you can't clear deleterious mutations as effective. I should say you can't clear deleterious alleles as efficiently. And because you have a small population, you have inbreeding, you're more likely to end up homozygous. So you end up with a lot of harmful traits that are recessive, but they end up being expressed in the population because you have a loss of genetic diversity and a loss of heterozygosity within that population. So it's called, over time, over generations, you see the fitness decline and decline and decline as the genetic diversity declines and declines and declines. You get a lot of homozygosity, and the population ultimately goes extinct, right? That's an extinction vortex or mutational meltdown. Now, what they turn around and do right here, this is the principle of using mutation accumulation to decrease fitness, useful in fighting viruses. That is different. That is opposite. This is error catastrophe. This is where you're increasing the mutation rate to increase the genetic diversity, breaking a bunch of genes by introducing new harmful mutations into those alleles. Now, it's the complete opposite in terms of what's going on at the population genetics level.
Now you've never done any work in trying to induce error catastrophe, have you?
Funny you should ask that. I actually did in grad school. That was like half of my dissertation was on that exact thing. Yeah.
Did you get error catastrophe to happen?
Turns out no. I couldn't.
Weird.
I couldn't. What I should say is I couldn't demonstrate it mathematically. I could not. I drove my populations to extinction. I could not demonstrate that it was due to mutation accumulation.
So, despite trying pretty hard, as I imagine, you couldn't actually demonstrate the actual occurrence of error catastrophe.
Yeah, I'll note here that this citation, these two citations are both wrong. Crotty et al. 2001. I will warn everybody that this is my field. This is my exact specialty: mutation accumulation, fitness, error catastrophe. That is my thing. So I know all these references. So Crotty 2001. This was a study looking at the effects of ribavirin, which is a nucleoside analog. It's a base analog in your DNA or RNA, and it replaces a nucleotide and causes mutations. So the theory and what they did in that experiment is that you could mutagenize a population of viruses and drive them to extinction over generations using this mutagen. The problem is that as a base analog, nucleotides are involved in lots of things in biology. So you don't just increase the mutation rate, you also mess with things like intracellular signaling and energetics in the cell, metabolic processes. You end up messing with a whole lot of things. And that same team actually published a paper a few years later describing 5 potential avenues of activity for ribavirin. So in that 2001 study, you cannot attribute those findings to mutation accumulation. There's other things they didn't control for. And then Carter and Sanford 2012, that's my favorite bad paper. That's the H1N1 paper, where they said that the fitness of H1N1 decreased from 1918 to 2009, at which point they claim it went extinct. They didn't measure fitness. They did the wrong comparisons in terms of what sequences they compared. So they just did not measure the mutation accumulation correctly. And also that lineage did not go extinct in 2009. So
didn't they use virulence as a proxy for fitness?
Strictly speaking, they used excess mortality among younger people as the proxy for fitness.
Yeah, because killing your host is always the best bet for a parasite.
Yeah, because they were looking at the 1918 influenza, they based their data on I forget the paper, but it was the data set that they used was excess mortality, and they show of the younger demographic, like the 18 to 30 something, maybe whatever the year range was, excess mortality in that compared to over 65. And the 1918 pandemic famously was very dangerous for young healthy people, but that dropped off very quickly into the 1920s.
And I think this is an example of, once again, not realizing why we define fitness the way we do. Because the thing is, you might think that the job of a virus is to kill its host, but the virus, to anthropomorphize it inappropriately, doesn't think that.
Yep. It's all about just who is the best competitor in that specific environmental context. So I want to move on from this, but the idea here is they're saying two things. They're saying, here's a thing that's well documented, and here are some examples of it with mutation accumulation, when this half right here is the opposite of this. They are separate things. They are opposites. And the authors are conflating them as though they are the same thing, and they are not. And John Sanford does that in his book, Genetic Entropy. Same problem. It's not good.
It's okay to kill your host if you're one of those parasitic wasps that lays eggs in a cockroach, and then it dies. Then it's okay.
Yeah,
That's not a
that's
virus,
true.
though.
So let's keep going here. Just stop me if you all have anything. Dakota, how are you doing? Welcome back. I'm glad you made it back to us. If you have anything to any thoughts, go for it.
Not at the moment.
Okie dokie. So let's see. We've got, we're just getting some definitions here. Okay, this is fun. For asexually reproducing populations, this loss is irreversible. This is talking about the loss of fitness due to mutation accumulation. Essentially, they're talking about Muller's ratchet here. For asexual populations, this loss is irreversible. No. It's not true. And it's demonstrably not true. We could do this in the lab and show, like, if this was the case, then the Lenski experiment should have gone extinct, right? Because those are asexual populations that are accumulating mutations. They should be dead, according to this statement. And, like, their fitness actually increased by a lot.
Yeah.
Yeah.
And
it's just wrong. This is just a
the
wrong
other
statement.
thing is, I don't remember if this paper does it specifically, but creationists like to then bring in Muller's ratchet, and I think they don't do it in this paper. But they then like to apply it to things like humans.
I have bad news for you.
Oh, they do it?
Look right below the part I highlighted.
Oh, goodness. Of course. Okay, guys. Okay, this is... I'm directing it
that's a problem.
I'm directing this at Sal, and I'm trying to be... Look, I very easily fall into being a snarky dick, and I don't want to do that, because I know that that's not the appropriate thing to do right now. And I like Sal, and I don't think he's being intentionally misleading here. But look, Sal, you cannot take a thing that was maybe thought to be a problem like 50 years ago, and that now no one really considers a problem, and that even when it was considered a potential problem, was only a problem for asexual organisms, especially asexual organisms that couldn't do recombination, which many of them can anyway, and then apply it to diploid animals. Like, you can't do that.
And I want to just say it wasn't even proposed as a problem. It was the whole framing is wrong, because Muller's ratchet was never proposed as a problem. It was proposed as a selective pressure that would favor sexual recombination.
Yeah,
like that's the reason for it, because you look at sexual versus asexual. There's a lot of costs associated with sexual reproduction.
You gotta find a mate. That in and of itself is a pretty big cost.
Dangerous, right? You got these showy tail feathers, make it hard to fly, make it impossible to hide from predators. Like, stuff is dangerous, right?
Look, just so last time I went to look for a mate, I got attacked by a lion. Not
cool. Were there lions back then?
Well, I mean, the last time I looked for a mate, sure. Look, I've got a whole 150 million year history. A lot of it includes lions.
Okay, fair enough. I'm not up on when cats speciated. I don't know when.
I mean, it's way after the Jurassic when I was hatched. But like I said, I've got the whole 150 million years of work with.
It's a bit, everybody. He's a dinosaur. It's a bit.
Yeah.
So Muller's ratchet, quick overview on Muller's ratchet. The idea is that sexual reproduction has a cost to it. So there needs to be a strong selective pressure to favor it over asexual reproduction. And Muller came up with this idea that, well, recombination is a really useful thing to do, because in the absence of recombination, if you have a good mutation, you have a mutation that's helpful and that's going to be selected for, well, you might have a bunch of other mutations present in that same gene, allele, chromosome, whatever, that are bad. And if you select for the good one, you're going to drag all the bad ones along with you, and that's going to be a drag on your fitness. But if you could recombine, if you could use sexual recombination and break up those mutations, maybe I got good mutation here, bad mutation here. Well, what if I swap? I break the DNA in between those two, I swap with somebody else. Now I've separated the good from the bad. I can keep the good and select against the bad. That's a major benefit.
Yeah.
And that only comes with some form of recombination, sexual or otherwise.
It's sort of like taking the linkage block down from whole genome.
Yeah, exactly.
Like that whole genome linkage blocking is linkage blocking is not the greatest thing.
That's the problem, right? So what the idea of Muller's ratchet is, this is a potential issue that would impose a selective pressure for sexual reproduction. That's the idea of Muller's ratchet. What creationists do is treat this as an actual problem for evolution, as like, no, these mutations are actually going to accumulate. Like, no, it was never an empirical observation. It was a proposed explanation for like the selective pressure underlying a thing, right? That's Muller's ratchet. That's the idea.
Would that be sort of like when you have a beneficial mutation of I forget the name of it, but when it gives you a trait where you have a resistance to malaria, but you don't have the side effects of sickle cell anemia?
No. So it's not
trait.
the same thing as that, because that's the exact same mutation that has two different traits associated with it. So that's called antagonistic pleiotropy, where you have a specific allele that has both a fitness good effect and a detrimental effect on your fitness, right? So it's one allele, one genetic trait that has multiple phenotypic traits with opposite effects.
I'd also like to point out that the beneficial phenotype associated with sickle cell anemia in malaria-heavy areas actually requires Muller's ratchet not to be in operation, because it requires you to be heterozygous. So it actually requires that you have these non-genome-wide linkage blocks, so that way one parent can give you the allele for sickle cell, and the other one can give you the normal allele for regular shaped blood cells, and therefore you have the intermediate condition in your red blood cells, so they can still sufficiently carry oxygen, but also you have resistance against malaria. And so just that in and of itself, it says Muller's ratchet is already out the window a long time ago for this particular phenotype.
I do want to say, strictly speaking, that's not linkage blocks, but the broader point remains that that's...
Well, I mean, for Muller's ratchet, you basically have to have whole genome linkage blocks, but obviously we don't in this case. So that's why it's already out.
The other thing here is to talk about genetic load, and like that's from before like neutral theory existed. So that's a problem, because once 90% of the genome doesn't matter in terms of like what its sequence is, then like mutation accumulation becomes a much less serious issue for you. So it's just worth noting that like we need some context here. This is from 1950. It's by 1980. This is not a thing. This is wrong by 1980, and now it's 2021, and it's still wrong.
This is ever so slightly off topic, but it's something that I would like to see addressed by Yarnock Creationists, and I suspect if you will watch, I would like for you guys to explain why it is that neutral variation also follows the nested hierarchy that we established from non-neutral variation as well as morphology.
Yeah,
because there's no good reason for it to, unless evolution is true. So I would love to see a rigorous paper about that from creationists.
Before we move on, I just want to point out one funny tidbit here. So talking about extinction vortex and extinction in small populations, if you put Noah's family into Mendel's accountant, it goes extinct.
How fast?
Fast enough that we don't get to 2021.
There's no surprise there. Yeah.
So like people have done that and they actually go extinct. So I just want to point that out. According to the model that they will later say is the best model.
Also remember, Noah's family had an effective population size of at maximum 5, not 8. Because and 3 of them are 5. Full siblings. 5. Max 5.
Dan, wherever the danger was, Noah's family was somewhere else.
They were somewhere else. Yeah, there was, and that's true for genetic danger too.
The whole thing is miraculous. So I guess maybe
say that.
Yeah, just say that, and then.
But then don't try to make the
halls of
gen
science work.
Right. Just call yes, it was a miraculous event, fine. But then don't try to make the population genetics work. And if you do try to make the population genetics work, I'm going to point out that it's ridiculous.
Yeah.
This is just not what?
Nope.
What? Why? No. It's just not we could do experiments. We could do mice exist. Mice haven't gone extinct from mutation accumulation.
Yeah, their generation time is a few weeks, basically.
If Sanford is right, mice should be extinct. You can do the math on that. It's a mammal. It's got a genome that's in the same order of magnitude as humans. I forget what their genome is. It doesn't matter specifically, but you can do the math on it. If Sanford is right, mice shouldn't exist.
Interestingly, mice are actually pretty closely related to humans because in the spectrum of mammal diversity, rodents and lagomorphs, the rabbits and hares and whatnot, are the sister group to basically primates plus colugos, which are a weird sort of gliding animal that no one knows about, but they're cool. So there's a reason that we use rodents as model animals for human like medical testing and whatnot. It's because we're related pretty closely.
Yeah.
And I think the long-term E. coli experiment wouldn't be going for so long if this was true
Oh,
either.
not at all. Oh, the E. coli experiment should have been gone years ago.
Yeah. Oh, and we'll get to that. That's in here. And I think we've decided we're just going to do this thing. So what page are we on? We're page 28. We're making progress. Okay.
Yeah.
Before we get any further, I just want to define quasi-species, because this is a really important concept in evolutionary biology that's very frequently misunderstood. And it's important for all of what we're talking about here, because the arguments that Basener and everybody are making are basically that you are going to have a decline rather than equilibrium between mutations and selection. And a quasi-species is an example of what they are ignoring, basically. Even though they bring it up, they basically ignore that it's a problem for their argument. Because the argument is that you don't maximize fitness, right? Fisher is wrong. Therefore, you decline rather than have an equilibrium. Well, a quasi-species is an example of an equilibrium that is below the maximum fitness, because what happens in a quasi-species is where the mutation rate is such that the most fit genotype is not the most common genotype, which is a weird, just think about this for a minute. So what you have is you have a bunch of genotypes. They have different levels of fitness, right? And we're taking a snapshot. We're not worried about like fitness then, fitness later. We're just snapshot right now. You have a bunch of genotypes, and then we're going to have different levels of fitness. The one that is the best is going to be selected for. But because of the high mutation rate, you're constantly mutating away from that. So what you end up with is a population that has, instead of the most fit genotype being the most common, you have a pool of closely related genotypes that fluctuate at equilibrium with each other that are within a few mutations of that most fit genotype, because you have mutations constantly moving you away from it, and you have selection constantly selecting for it, right? So you have, and these two things are at equilibrium, where you end up, instead of having a distribution that looks like this, where you have a defined peak where the most common is the most fit, you end up with a very flat, wide distribution where you have a lot of things that are very similar frequency that are all genetically very similar to each other. So that's a quasi-species.
Dan, are you saying that it's already well established within population genetics that the most fit genotype will not always be the thing that the population arrives at?
Turns out that is a well-established thing. So talking about all this stuff with fitness maximization is kind of silly, because like nobody thinks that's a thing to expect. There's not, you wouldn't expect that in tons of situations. Yeah, it's a well documented thing. You could just show that like viruses, like RNA viruses, exist in this state, right? Because they just have high mutation rates. Anybody, any Deadly, Atheist Junior, any other thoughts as we go through? We got some math here. We got someone pointed out earlier that we have a pentagram. So I don't know who drew this, but whoever it is, I see you and I appreciate you.
I'm just gonna say that I'm with Dan on this one, where it's like the math isn't really my problem with this, and also I'll admit I'm probably not very well equipped to analyze the math on a pure mathematics standpoint. Like the limit of the math that I do normally is like n-dimensional vector math, which is, it sounds fancy, but it's really not fancy. It's not even calculus fancy. So like, yeah, I have no problem accepting.
Pentagram. I'm sorry, I said the wrong thing. I'm sorry.
We're okay. Well, it's time to crucify Dan for getting the geometry wrong. But
sorry.
I'm Dan on this one, where it's just like my problems here are before the math starts.
Yeah.
And if your premises for your math are wrong, your math can be as accurate as possible. And I'm willing to just say, yeah, sure, the math might just be completely fine. Doesn't matter.
This paragraph, so we've jumped ahead a little bit, but this is in the same vein. This is talking about the extinction threshold, where you have a mutation rate that is sufficiently high that they'll accumulate infinitely within a population, drive it to extinction. Another word for this is error catastrophe, is like what that process is called. Here's the thing. It is at best an open question whether this is a thing that can actually happen in a population. It is often claimed that it has been demonstrated, and like I have a list of like 15 papers that people will point to and say, there it is, error catastrophe. And I can go through each one of them and tell you why it's not. Some of them, it's a problem with the math. Some of them, it's like the one I mentioned earlier, the Karate 2001 paper. They just didn't control for other effects of the mutagen that they used. Like, we can go through that. I'm not going to do it here, but I could do that another time at some point, and just like talk about the creation myth that like error catastrophe is a thing that has actually been observed. That is not the case.
I have a real quick question. Is it fair to say that error catastrophe is just what scientists call, I guess, evolution or
genetic entropy?
Genetic entropy. Yeah. So genetic entropy is just the pseudoscience version of
the math. The math of genetic entropy is error catastrophe.
Okay.
Yeah.
So those terms are basically synonymous.
It close. I think Sanford adds a little bit of baggage by saying like this is a universal thing that has to happen, particularly in humans, right? So I think the genetic entropy hypothesis is broader than just like the mathematical thing that we can document error catastrophe,
Okay.
right? But error catastrophe, the mechanism, is what underlies genetic entropy. What Sanford essentially argues is that error catastrophe, far from being something that we can potentially induce with mutagens, is something that is occurring right now in everything, basically. Not quite everything.
The thing that always gets me about that is like, look, we have populations that reproduce like orders of magnitude faster than humans. Why do they exist?
I mean,
Period.
there's experimental evidence that this statement is wrong. Like that this is not a thing that exists. And the citation on this is Springman et al. 2010. J.J. Bull is the senior author. They mutagenized a population of viruses, expecting them to go extinct. The mutagenesis was such that they induced every mutation many times over. So they were mutation saturated, right?
Which humans are too, right?
I'm sorry?
Humans are also mutation saturated.
The human population has sampled every mutation many times over because there's a diploid human genome has 6 billion bases, and each person has approximately 100 new mutations compared to their parents. And there's 7 billion people, so you just do that math. We have sampled every mutation a lot, right? And when you get a population in that state, according to John Sanford, and this is the genetic entropy argument, you can't select for the best. Even if you select for the best genotypes, everybody is in decline because almost all mutations are bad. Virtually no mutations are good. You can't select for the good ones anyway. That's part of his argument. So the population must inevitably go extinct. That's genetic entropy. Well, if you can demonstrate that experimentally, that you can mutagenize an entire population to the point where every mutation has occurred multiple times in that population, then you've disproven Sanford's hypothesis. You have shown that it is not valid, like just straight up. And that's been done. And for good measure, we're confusing error catastrophe and mutation meltdown again. Again, these are opposites. This and this, extinction threshold, mutational meltdown, those are opposites. They are the 180-degree opposites from each other.
It's just like hypermutating inbreeding.
Yeah, exactly. It's the same magnitude of error. Yeah, it's the same thing. I got another super chat here from Carrie Bronson again. Thank you. Genetic entropy is catastrophic with errors. I don't think Sanford. Yes, that is true. It is it is catastrophic with errors. Yes,
that's awesome.
Okay. Let's march on. And again, if anyone sees anything that jumped out at them, stop me. Okay, so I just want to point out that this paper, Basener and Sanford 2018, it's based on the same, DFE is distribution of fitness effects. Basically, if you plot out the fitness effects of mutations across your x-axis with the frequency on the y-axis, that's your distribution of fitness effects. And this paper is based on the same one that Sanford uses in Genetic Entropy and the Mystery of the Genome, and it's made up. It's not real, it's not empirical, it's not based on observations. It's Sanford modifying the parameters for a model, and it's actually Kimura's model from the 1970s. It's not actually real. What? My dog is joining us.
Yeah, the dogs here are trying to interrupt us here.
There we go. Okay, so that's what's going on there. Yeah. Okay, so let's keep going.
We need to just have a dog stream. We're just the dogs stream.
That would be great. So this is a nice illustration of this kind of quasi-species thing that I talked about earlier, just showing how like this is the best one, but you're going to constantly mutate away from it, and at a certain mutation rate, it flattens the distribution, right? So I actually like that figure a lot.
It might be the mode, but it's not the majority.
Yes, maybe it may not even be the modal one, even if it's not majority. It may be basically the actual phrase for it is really cool. Instead of survival of the fittest, they call it survival of the flattest, because it's about finding genotypes that have a number of closely related genotypes of approximately equal fitness. That's actually going to outcompete populations that find a very high fitness genotype, because you're likely to have that's likely to be less stable mutationally.
Right.
So what you're selecting for, this is way in the weeds, but what you're selecting for is a trait called mutational robustness, meaning you're actually selecting to have a situation where mutations are unlikely to negatively affect your fitness. And yes, that's a phenotype you can select for.
Yeah. One thing that I think is a way you can picture this, if you're familiar with a particular model, which is random walk and fitness landscapes. So you have a population that's taking random walks on a fitness landscape. Any time that population reaches a peak on the fitness landscape, the random walk is all going to move them away from that peak. So that population is going to spend a lot of time with the majority of them not on the peak, because where else can you go when you're taking a random walk? Be away from the peak.
So you're going to end up selecting for a very flat landscape where there's not cost to walking away from the peak.
Right. And
which is so
now,
cool.
Of course, that depends on knowledge of a fairly complicated model for fitness, but
yeah, it's really cool, fun math that happens in viruses. It's so cool. Okay, so the mean effective mutation is generically negative. I don't know if that was supposed to be generally, but like whatever. Point is, you need a citation on this because most mutations are demonstrably neutral.
Generically makes no sense.
Like they are new. Like most mutations don't have an effect on fitness. That's just how it is. Like you could quantify that.
Okay, so I want to point out something from Sal's live stream where he was presenting his thoughts on this. So one of the big points he made about this was that he's taking very seriously the idea that 80+% of the genome is functional on the basis of ENCODE. Now, I know
get to it.
Okay,
we're going to get there.
I think that I don't want to say that Sal is definitely speaking for the authors, but I think that because he is an author, and I'm sure they discussed this beyond just what's in here, that this was in the minds of more than just Sal. Mm-hmm. And I know we're getting into it, but it's just not true that 80% of the genome is functional in the sense that it changes to it has effects on the fitness of an organism. If you want to define functional in the broadest sense of like does anything anywhere in the organism, okay,
has activity.
Right, but spurious transcription of a few codons doesn't affect the overall.
It's super common. Yeah. I mean, some of we'll go into this when we get to ENCODE, but some of this stuff is like it's quantifiable, countable numbers, like single-digit numbers of RNAs. Like that's the level of transcription you're talking about. That's not going to do anything. It's just not. It's not functional if you're making 5 of them.
Right. I mean, and the thing is, it's not even usually resulting in transcription all the way to like getting a protein out of it. It's just like, oh, hey, some RNA got produced, and then it did nothing.
Yeah.
Okay, sure. I don't disagree that this happened, but like, who cares?
So here's a couple things I want to point out right here. Harmful mutations outnumber beneficial ones. True, but the majority are selectively neutral, and that's an important distinction. And then right here, this is again, we're doing this again with the beneficial mutations. It's almost the wording is almost exactly the same. So rare they're statistically undetectable. That's the same wording that Sanford always uses. That's wrong. It's dishonest.
And it's not what Cordova said.
Right. Here's the DFE, the distribution of fitness effects. So you've got zero, like precisely neutral, and then you're going to have a zone where it's effectively neutral around that. And what they show is this distribution for harmful, and then over here, essentially zero and basically zero selectable beneficial mutations.
Right.
This is wrong. This is not correct.
It is wrong, but it's also the graph that you get if you look at Kimura, because this is the graph that Kimura uses, but because he explicitly tells you in the paper, hey,
Why?
I know this is not correct, but I have to use this distribution in order to model neutral variation, because if I put anything to the right of zero, it ruins the signal I'm looking for.
It's so much worse than that. If I remember the paper correctly, I should actually pull it up right now and check, because if I remember correctly, I actually think he doesn't even draw the positive side of the graph.
Oh, I don't remember, but that might be the
only
case.
draws the negative side, if I remember. Oh, hang on, my screen. There we go.
So Robert Monreal asks, "What is the
yes?
X
He only
axis of the graph?"
he only
So
draws.
that's-
I can confirm he only draws the negative side of the
Okay.
graph. Kimura doesn't even draw. It's in Kimura 1979 Figure 1. He doesn't
only
draw the
draws the
positive
left
side
hand.
of the graph.
Okay.
He
So-
only shows the negative side.
I want to answer Robert Monreal, and if I'm mispronouncing your name, the x-axis of the graph is change in fitness, which is be relative fitness. So
Relative fitness. Yep.
So the average fitness of the population is going to be defined as zero, and these mutations that you're seeing is this many mutations are going to result in a whatever change in relative fitness compared to the population as a whole.
Yeah. So, like, if you have a mutation over here, that means that the individuals with that mutation are 10% higher fitness than the average for the population, or whatever baseline you're comparing them against.
So,
right?
if the normal,
that's what that means.
If, say, the normal number of grandchildren that reproduce is 20, and you have that mutation that puts you at 0.1, well, you're going to have 2 grandchildren reproduce
on average.
On average, right?
Yeah.
Which, congrats. Which I guess actually makes one of my.
I mean, that's a big. That's
really
actually a
big.
big difference. That's a huge difference. Very few single mutations are going to generate that level of.
Oh yeah, it would
increase. That
enormous. Would be amazing.
Yeah. But so that's a problem.
That would be like immunity to the current horrible plague kind of levels.
Yeah. You can't. So the point is, you can't like base your math on this. This is not empirical. It's not correct. This is not. Also, you can't not show the neutral range in here, because there's a range around 0 where it's too small a difference to have a detectable effect, so it won't be selected for or against. That range is not set in stone. It's going to change based on things like population size. You have to include that. One of Sanford's major errors is he assumes a constant distribution of fitness effects, basically saying that this does not change with population growth, with population reduction, with changes in the environment, changes in selective pressure. This does not change, according to Sanford, and that's wrong. It's just wrong.
Mm-hmm.
Let's see. Continue on. Okay, this is just me. You don't need to worry about this. This is just me. This is just me commenting for me. It's population genetic stuff. It doesn't matter for the big picture. Okay, I'm being nitpicky, but like very small deleterious. It's very slightly deleterious mutations. The S is slightly, not small. And this is the genetic entropy argument is what they're doing here. They're not saying it, but this is the accumulation of very slightly deleterious mutations. This is the genetic entropy argument, right? That these mutations occur, they hurt fitness, but you can't select against them, right? That's what this argument is.
Would I be accurate to say that they seem to be repeating themselves a lot in this paper?
Yes, I think so.
Okay.
Which, I mean, they're setting up the argument. They're being very methodical about it. So I think, I think it's okay. It's just, yes, okay. It is repetitive at times. Yeah. There were places where I'm highlighting things I thought I highlighted this already. And it turns out it's in like multiple places. It's Fisher's fundamental theorem fell out of favor. Yes, because it's not fundamental. Okay, let's see. This is a whopper. The more realistic the parameters, the more certain the net decline. It's not true. It's not true. You know what has the most realistic parameters? Real life.
Yeah.
And in the experiments, we can mutagenize populations, or we can just let them go. And in neither case do they decline on net. We can show that. Like, I don't know what to tell you.
Yeah,
That's
it's
just-
I,
that's
I
just not
forget
true.
I forget the exact catchphrase, but it's something like, you can basically theorize about something 100 times, but one experiment is worth a lot more than that.
Yes. Right. Yeah, you can just, like, this statement, you can just, it's wrong. It's just
Yeah.
factually wrong. Okay. Okay, now this is where now I'm getting snarky, because, like, we're going to talk about how you need to have really realistic things, and then we get into Mendel's accountant is, of course, the one that we're going to bring up, which no. I want to direct some people to some threads on the Peaceful Science Forum, which is a really delightful place to talk about science. It's a really nice place to talk with a lot of, like, actually working scientists in a lot of different fields. Very cool place. People have dug into Mendel's accountant, tried to get it to match actual experiments, and couldn't do it, which is a problem.
Yeah, that's what you might call problematic in academic speak, which, as I pointed out before, basically means absolutely insanely bad for your proposed idea.
Like this right here, like, you put in the parameters of the Lenski experiment. This is the Lenski long-term evolution experiment with E. coli. That's been going for decades now, tens of thousands of generations of E. coli, lots of mutations accumulating. They've documented fitness, throughout the process.
Mm-hmm.
If you model that in Mendel's accountant, it should go extinct. But, like, the real life, what ATH Jr. just said, the experiment is worth more than the model, right? Like, I don't need to guess what's going to happen. I don't need to model what's going to happen in that situation, because we've done the experiment since 1988.
Right.
It's still going. They're not extinct.
It's almost like if you make a model to confirm your predictions and then it confirms your predictions, that doesn't really mean anything.
Yeah. So I do want just programming note here. Let's see. We're on page 44. We're going to finish this by 11. Okay. We're going to finish this by 11. We're going to get through the whole paper. That means we might have to speed up a little bit, but that's okay. H1N1, I don't need to beat this paper to death anymore.
Yeah, we already talked about it.
The fire H1N1 paper so bad.
So it said up there, like, the more realistic the parameters. Like, I feel like they just said that in, like, the page before, like, almost the exact same thing.
Here's where Neff, before he left, pointed this out, that we're just, like, hitting all the high notes here, like waiting time problem. The paper they cite here from 2018, I've read that paper. They left out recombination. They actually say in the paper that recombination wouldn't affect their math. I'm sorry.
Excuse me?
Your math is about how long it takes to get two different things together in the same genome. Like,
yeah,
recombination is going to affect your math. That's important.
It might be the primary driver in the waiting time for.
Well, here's the thing. They model asexual haploids in this paper. But then you want to apply it to things like humans and our divergence from our common ancestor with chimps. But if you're basing your math on asexual haploids, I feel like that's not going to work, because humans are diploid and reproduce sexually.
It won't even work on asexual diploids. Even there it will work.
Yeah.
Because, spoiler alert, there are plenty of asexual diploids out there, and they recombine their own genome to make sure that their offspring aren't actual clones.
There are even some asexual tetraploids. It's awesome. It's really cool stuff. But also, strictly speaking, that's not even true with no recombination. That's strictly not even true for asexual haploids, because they have other ways of doing recombination. Like, bacteria are really good at doing recombination that is not sexual recombination, right? It's not recombination tied to sexual reproduction. It's different things, but there's mechanisms to do it, and they do it a lot.
It's almost like the existence of asexual reproduction to this day means that this idea can't be true.
Right. It's and creationists acknowledge that bacteria do that, because when they try to undermine phylogenetics by talking about how, like, it's messy because you've got all horizontal gene transfer. Yeah, that's recombination happening. among the prokaryotes. Like so this is not a real thing. Okay. So this section, I'm just gonna spoil it. They're gonna take a bunch of stuff out of context right here. Like they just, Muller, Kimura, Kondrashov, Moran, Lewinton, like they hit everybody, right? There's a bunch of quotes here. It's just basically saying you don't get fitness maximization. Fine.
Which true.
There's quotes. Now here is where I want to land on ENCODE for a second, because they're citing this seriously as this 80 to 100% functionality in ENCODE, which is like, this is not, I'm not going to do anything except laugh at this. Like, this is not a serious argument. Even ENCODE doesn't buy this. That was the preliminary number from the 2012 ENCODE paper. They published a number of follow-up papers, most importantly, the 2014 one, where they went back and revised this downwards, and they were extremely specific and extremely careful about the way they explained it. And it's in that follow-up 2014 paper, which they do not cite, where they talk about how, there's a lot of transcription, but a lot of it is very low levels of transcription. So instead of just measuring transcribed or not, they measure the level of transcription. They can document a lot of transcription versus very little. And they go out of their way in the 2014 paper to say, it's probably the case that a lot of this low-level transcription is spurious. The amount of transcription that's having an effect that's actually selected to do a function is a lot less than what we originally thought. Like, they go through that, and they go through other things like protein binding, and they do the same thing.
In 2012, didn't they also look at methylation targeting as if it's targeted for methylation, it's active?
Yes, they
Which
did
is sort of the opposite of what people normally think of. Because if you're targeting a part of the genome for methylation, that makes it harder to transcribe. It makes it less likely that that part of the genome is going to be expressed.
Bingo.
So to call that functional 100%. Is a little weird.
I think they have the causality backwards when it comes to it, to the extent that methylation targets are specific. I think they have it backwards. It's not that this sequence is specific and it's functional because you need that sequence for methylation. It's that we adapted to recognize things like viral insertions or transposon insertions that were bad, that we wanted to inactivate and prevent them from mobilizing in our genome. So we adapted to methylate the poop out of them. So they crunched up in really dense heterochromatin so that they couldn't transcribe, they couldn't mobilize. That doesn't mean that sequence that's highly methylated is functional. That's the thing in your basement that you're trying to pack into the box as efficient. You're playing Tetris with the crates in your basement to try to stick it as densely as possible and then shove it in a corner so you don't trip over it when you're doing your laundry, right? That's what the methylation is doing. Mm-hmm. That's not telling you it's functional. It's the opposite of that.
Right.
We could do a whole 2 hours just on ENCODE and bad interpretations of ENCODE.
Maybe
but I want to move on.
We should sometime.
Maybe we should. So we should do that sometime. That'd be fun. We should talk about Dan Grauer here and ENCODE. Because he goes and talks about where is it? Here we go. He talks about how the authors here talk about how Grauer said if ENCODE is right, then evolution is wrong. What Grauer meant by that. And I've talked to Grauer about this. So what he meant by that was that if ENCODE is right, then we need to rethink some of what we know about population genetics, right? Because clearly something is not matching up here. What creationists take him to say is that it's entirely wrong. Evolution, that is, which is
universal common ancestry is completely debunked, apparently.
Well, you got to get that quote mine in there. You have to get that quote if you're going to talk about
that's
ENCODE.
One of the classics.
Yeah. I mean,
it's not the darn in the eye, but it's up there. It's top 5.
Yeah.
So I want to jump in here.
I want to say to Sal while you scroll down, Sal.
Yeah.
You put your name on this, man, and it has a bunch of quote mines, and I think too highly of you for you to be doing all these quote mines. I really, 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 really need you to address this at some point. Why is it that you're quoting numerous authors in this paper? I'm going to say you because, you signed off on it, who are actually saying basically the opposite of what you're implying them to say. It's not okay, man. Please put something out that addresses this. Because I really don't want to think of Sal as the guy who lies about what people say. So maybe you didn't check on it. I don't know, but you got to say something about it, man. Sorry.
I'm just going to put in the chat here. This is the link to the 2014 ENCODE follow-up. I just put that in the chat, and the relevant figure there is figure. Just scroll into it. It's figure 2 is the relevant figure where they look at things like the level of transcription rather than just does it exist or not.
Yeah.
Okay, so
it's interesting how rarely creationists look at follow-ups to papers that they quote.
Yeah,
because it's basically
true.
never.
And that's a perfect segue. There's a perfect segue, sir.
Nice.
Because we were just talking about Grauer, and he had a paper in 2015 that creationists love to cite about how in order to have mutation-free offspring, you would need to have bajillions of offspring, right? Like tens of thousands or more, right, per reproducing couple. And then you could have, based on mutation rate, the functional fraction of the genome, like you need to have a ton of offspring.
Which was a big talking point when Sal presented this paper on his channel.
I'm sure. So there's two problems here. There's a theoretical problem, like a conceptual problem, and then there's an empirical problem. So the conceptual problem is that mutation free is not what selection is going to get you, right? What you're talking about with selection, assuming whatever, right, on however you want to assume selection is operating, what you're going to get is a fitness, right? But that doesn't mean mutation free. It just means you're maintaining a viable population. There's still going to be mutations happening in individuals in that population that are going to be transmitted generation over generation. Now, on net in the population, you may not be getting any new mutations, right? Because like humans, given our genome size, mutation rate, and population size, we're not going to find any new point mutations at this point, right? We're all experiencing everything that's possible.
We've covered it already, yeah.
We've covered it, right? But individually, you're still going to have mutations happening. You're not going to select for a mutation-free individual. You're going to select for the viable individuals, right? So that's the first thing. The second thing, I guess there's three things. The second thing is just a problem with the math right here, where it's you need to eliminate X number to maintain a population of 100 given the distribution of fitness effects. This is ignoring neutral mutations. It's just assuming that if you have a mutation, then it is harmful and must be eliminated from the population. Neutral mutations are a thing. Like, you can't just ignore that.
As far as I know, they're most of mutations.
They are most, funny enough. The big problem, though, the big problem right here is that they cite, this is like the empirical issue that I have right here, is they cite this Grauer 2016 paper. There are two additional things they don't cite here. One is the correction to that paper, because the numbers they cite are from the uncorrected paper. But Grauer, in 2020, it was pointed out to him that his math was wrong. And he went back and looked and said, oh my, you're right, my math was in fact wrong. And they appended a correction to the published paper. So if you go and look up this paper right now, it has a correction on it that fixes Grauer's math. They are citing the uncorrected numbers. Now, the reason Grauer went and did that is that there was a 2020 paper by Benjamin Galeota-Sprung. That's hyphenated. Galeota-Sprung is the lead author. 2020. It's called the functional fraction of the human genome, I think is what it's called. And they actually did the math correctly for Grauer. And Grauer was a reviewer on that paper. And I've actually emailed back and forth with Grauer about this. So like I've gotten the story straight from Grauer here. He was a reviewer on the paper that fixed his math. And he was like, we all agree they are correct. They did it correctly. And when you do it correctly, these numbers don't hold. This table does not hold if you do the math correctly. So
in most cases, it comes down to like you need fewer than 2 offspring for the story.
It's like 1.8, 2.2. It's single digits.
Yeah. Now I have a question. Is it possible that this was submitted before the 2020 correction?
That Maybe that was so. I brought that up when I talked to Sal about genetic entropy way back in like last June. It was in the second round that we talked. I brought this that 2020 paper up. So I don't know how long this chapter's been in the works. It's possible, but I know I've brought it to Sal's attention. It's
because if that were the case, then I would give them a pass on this one, but that seems iffy at best.
Yeah. Okay. Let's keep going. So here you go. Atheist Junior, we have made it. I know we waited a long time. Why have we not died 100 times over?
That's a really good question.
Dakota's back. That is a good question. This very slightly deleterious mutations again. This is genetic entropy, right? So, Atheist Junior, what do you want to talk about here?
Oh, well, I was just going to mention, because earlier Dapper mentioned how instead of citing actual books and, papers, a lot of times the quotations are from like just internet sites that are full of quote mines. And I had to agree, because this quote at the bottom of the page here, I tried to Google that to find the entire quote, and I could not find a single website that didn't start with, the uncritical acceptance. Every single thing was formatted the same as this quote mine is. And it was a lot of creationist sites too. So I thought that was kind of funny.
It's this paper right here. Oh, is that a book? I think I tried. Oh, that might be a book. That might be hard to find.
Yeah.
That was up on page 50 or wherever we were here.
As someone with a history degree, I get citing books, but it always makes everything harder.
And I feel like a lot of the quotes in this paper start in the middle of the sentence, and that's usually not a good
when
look.
your quote starts with an ellipsis, it is kind of a red flag. Sometimes,
Where are we going here? Here it is. There it is. That's from a book. It's a little tricky to find that one. Okay, now this is where we get into the redefining fitness part of it. So population geneticists define fitness solely in terms of success. I want to call out the Salem hypothesis. If you're not familiar with that, look it up. It's basically engineers are predisposed to pseudoscience. We see that a lot in biology.
Actually, there's an upcoming debate involved exactly that thing. Because Dan, you're going to be on the 24th, I think, or the 25th, debating an engineer.
Yes. Oh, true. Good point. Yes, absolutely. So here is where they get to it, right? It can be asserted on a pragmatic level that fitness is most simply understood as total biological functionality in a given context. Now, putting aside the problems with that, and I said at the top what some of the problems with that are in terms of like just evolution, but this is not Lewontin's argument. He is not advocating for this, but it's made to seem that way. But if you go and look this up, it's not his argument. So I just want to make sure that's clear to everybody. We have reductive evolution, and we are going to get into this 2013 paper. This is a biggie. The point of this paper is about cycles of increase and then select it down, increase and then select it down. The point of this paper, and there are mechanisms that do things like that. Like you've got recombination, you've got horizontal gene transfer, you have duplications. And we know of instances of full genome duplications that do this. We know of allo and auto polyploidy in plants that are going to lead to genome duplications. The point is that you duplicate things and then you get rid of stuff you don't need, but stuff that acquires new functions and new complexity you keep. So the dominant form of evolution, as they say here, the dominant mode of evolution is reduction of your genome over time. But what the creationists tend to leave out of this is that it's well documented that that's interspersed with increases. And they're very specific processes and we can document them. Like, it's just a thing we can do. They're just kind of leaving that out. They treat it as though it's like, oh, we don't know. It must have just gotten no, we know, like, for example, in the evolution of vertebrates, there were 2 instances of full genome duplication.
Yeah.
It's a thing we can do. Yeah.
We actually know of relatively recent full genome duplications in some amphibians. So it's not even a thing that's done even in animals.
Yeah. Okay, let's see. Here we go. Reductive evolution of the protein coding level. Yeah, this is true. Like, this is true. This quote is accurate. This is our understanding of animal evolution. You're losing protein coding genes. But what they leave out is that it's a cyclic process where you gain stuff and then you select you chip away at it via selection and drift, and then you gain stuff and then chip away at it, and it goes back and forth. Let's see. Keep going here. Okay, so this is definitions. This was for me. This is not like, you don't have to worry about this. Let's see. Yeah, this is just ragging on Hössjer again. So now this is getting into the idea where we can't get complexity, right? So now again, that's just for me. Don't worry about it. We're getting near the end here, everybody. This is good. So here we go. This is the bait and switch that I mentioned at the beginning, where we're going to say, okay, mathematically, Hössjer is wrong. You don't always get increases in fitness. And then we should redefine fitness as complexity. And now we're going to turn around and try to show that you can't get complexity because of the math we already talked about. And, oh, here's some empirical basis for that. So this is the claim, right? There's no long-term evolutionary progress, which is progress, right? It's not a valid framing for evolution. You can't, like, evolution isn't about progress. It's not how it works.
Yeah.
It's about change in populations. This is what that is.
You have to assume some end goal. Orthogenesis has been rejected for a while now.
Yeah.
Please don't bring it back.
Now, I just want to point this out. I thought this was funny because we talk here, I'm just going to quote this. Hössjer's work was an attempt to establish laws in biology akin to those in physics and famously compared his fundamental theorem of natural selection to laws in thermodynamics. Hey, any of the three of you know of anyone who tried to make something in biology that was the equivalent to a law of thermodynamics?
Sanford? Something like that?
Maybe. It's not the guy who's an author on this chapter. Is it? Did he come up with an idea that he named after a law of thermodynamics?
Sanford?
So, John Sanford termed his idea genetic entropy. And in his book, he explicitly says, like, this is a fundamental law of biological systems akin to the second law of thermodynamics. So I'm calling it genetic entropy. Like, so calling out Hössjer for doing that, you could argue whether Hössjer was right or wrong. I personally think if you come up with something, you don't name it the fundamental thing. You let other people decide that your theorem is fundamental. You don't, that's maybe getting out over your skis a little bit.
Of hubris.
But Sanford calling him out for doing the exact thing that John Sanford did is pretty rich. And that's all. I just wanted to point that out. That part, I just thought that was funny.
I'm just wondering when we're going to get genetic heat death.
Well, that's when everything goes extinct. When everything goes extinct as a result of air catastrophe.
Okay.
So here, there is no consensus on what fitness is. Like, this is not true.
Yeah, no,
like,
that's a lie.
This is funny. I love this. The usual notion of equating fitness with reproductive rate is fraught with contradictions. Are E. coli such higher fitness than humans? Yes.
Yeah.
Yes.
What? Okay.
Why not?
I want to point something out. Okay, look. This is not just to self. This is to creationists in general. Creationism isn't science. People doing creationist work are not doing science. There is a scientific consensus on what fitness is, and until you start doing science, which involves things like not quote mining, not pretending that Mendel's accountant can actually predict real-world events when it absolutely not, things like that, you are not having the conversation that scientists are having. They have a consensus. It's try and change it after you start doing science. Cannot pretend it doesn't exist while you're still not doing science.
Yeah. So I'm just going to leave that one there. It's wrong. It's just wrong. And it's not a contradiction. And it's a weird human-centric view of all of this, which makes sense from creationists, but it doesn't hold water in evolutionary biology. Now,
Yeah.
this last part here before we wrap this up is on the long-term evolution experiment, and somebody in the audience is going to appreciate this and or lose their minds, or both. We're going to see how it goes. So first we're going to just say like what their goal was. I don't think their goal was to like show continuous evolution. I think their goal was just to document what happened. I don't know if they've specifically said to demonstrate that they were continuously evolving, but that's what has happened. But it's the next one that I really love about this because it shows they don't understand the experiment at all. The E. coli represent a population that effectively has an abundant supply of food.
By the way, you have citation needed, which is why, of course.
Oh, there you go. We got citation needed. Thank you.
Which everybody search Dapper Dino and citation needed in your GIF keyboards for Facebook and Twitter and whatnot. It'll show up, I promise.
Now, if you're not familiar with the way the long-term evolution experiment works, or LTEE, the way this works is the bacteria, the E. coli, are each day are put in a flask. In that flask is a liquid medium that has some amount of food, but it is an insufficient amount of food to last the entire day. That is the selective pressure that has been imposed on the E. coli in this experiment. They run out of food and have to compete before they are transplanted to the next flask of growth medium. That is the selective pressure that has been imposed on them. That is what drives the evolution of the E. coli, right? So to say they have an abundant supply of food is completely missing the point of the argument. They are very specifically, on purpose, resource limited in that experiment. They run out of food on purpose.
Otherwise, what would the experiment be?
The experiment wrong.
Like,
yeah,
like
it would
what would the
it would be stabilizing selection and drift. It wouldn't be adaptive selection to to this specific environment. Just wrong. Like, in the 4 authors, does no one know how the Lenski experiment worked?
That's possibly yes.
Because
honestly, that wouldn't shock me if none of them have actually looked into it enough to know how it works.
They run out of food every day. That's the point.
Well, except the citrate metabolizing ones.
Oh, you mean the one where they have unregulated continuous expression of the gene that regulates citrate uptake? Except that's not the case at all. Except that's not true. It's not unregulated continuous expression. It's regulated expression in 2 different environments. One of them is anaerobic. It's still regulated if there's no oxygen, but it's still regular, right? It's on if it's anaerobic, that the ancestral one. It's still regulated, and then that gene duplicated under the control of a promoter that was active under aerobic conditions. It's not unregulated. It's just operating in a different set of conditions. It's not unregulated.
I like how the paper says that they gained the ability to uptake the citrate, because they're definitely not going to say that they evolved the ability. They can't say that.
Right. And it's not citrate metabolism or aerobic citrate metabolism. It's all it is they gained the ability to uptake, which is not true. There were other mutations that needed to happen to have that the CYP plus line. There were a bunch of mutations that they documented, and some really cool epistasis going on in there.
So in the chat, CTR0 points out they wouldn't evolve citrate as a carbon source if there was an abundance of food.
Right.
LMAO. That's part of the point. Yeah, there's not enough food.
Yeah.
So some of them evolved to eat citrate when they shouldn't normally under normal circumstances. That's like the entire point of this whole thing.
Yeah, that's why it's cool. God, it's so bad, right? It's so bad. All right. The other thing is this is like a gotcha. Like, it increased rapidly and then slowed. Yes, because they're asexual, so they have clonal interference, which means the biggest effect mutations reach fixation first. And so it's going to go fast and then slow down.
Even if they were sexually reproducing and diploid and whatnot,
like that.
At a certain point, you're going to have a lot of relatively rapid adaptation because there's strong selective pressure. But as the selective pressure gets weaker, the rate of fitness increase is going to slow because the pressure is lower relative to your current fitness.
Better.
That's just how that's just how the math works in every case.
Yeah.
Like that's
So
I don't know.
yeah.
It's very
Yeah.
frustrating when it's just like the very basic like algebra level, like high school level math says, yes, this is what we expect. And then you say, haha, therefore you're wrong for expecting the thing that everyone expects.
Right.
No, that's not how any of this works.
Right. So this paragraph is really interesting down here. So what they point out is that 6 of the 12 populations have lost some DNA repair mechanisms, and at least CTRO can correct me on this, but it's at least 1, and I think actually 2 are what's called mutator phenotypes that have like way higher mutation rates. And if Sanford is correct,
is a phenotype you can select for.
Oh, isn't that weird, right? If Sanford is correct, then those 6 lines should just go extinct, right? Because they're just going to have way too many.
More mutation, more genetic entropy.
Right. So like those 6 lines should be extinct. The fact that they're not, and specifically the fact that the mutator phenotype ones are not extinct, that just completely invalidates genetic entropy. But here's something I want to point out here. This reference right here, Penacea 2013, downloaded that, I read it, are accumulating slightly deleterious mutations at a much higher rate than normal. That paper doesn't say that. It says they're accumulating mutations, but this slightly deleterious, that's a little bit of editorialization by our 4 authors of this chapter.
Yeah, that's just the imposition of their assumption that most mutations are slightly deleterious, which has not been established.
But that reference is not saying that, and you need someone to go into this experiment and show that that's the case before you can make that claim. That reference doesn't do that. I checked.
I want to say, go ahead and do it, because that would work.
That would be cool.
There's a lot of
do science.
A lot of times, young Earth creationists, and this came up when Dan, you and I were talking about that ridiculous fly paper that is on my channel that people should go watch. This is like, look, you've raised good questions. There are interesting experiments to be done here, so do them. Do the work. Stop pretending that you have when you haven't, and go do it.
Exactly. All right. So this is what we should expect, right? This is the gotcha with this one is, they lost some functionality. Yes, because they're adapting to a highly specific environment where they talk about, for example, that many of them have lost their flagella because the flasks are shaken. Like when you put these, if you've never done microbiology, this is not going to make any sense, but you put them in a shaker in an incubator and it moves the liquid around so the cells don't clump, so they stay suspended in the medium. And when you do that, you don't need to have flagella for the cells because they will move through the medium due to the shaking.
Well, the experiment is moving them for them, so why bother spending metabolic energy on
You can save a lot of energy by getting rid of your flagellum then, right? So that's cool. But this is the thing. And the authors get at it here. They get so close to getting it because they say, which turned out to be advantageous in shaker flasks. Yes,
which is where they are. Exactly.
Which means it's context dependent. It's not about functionality in an absolute sense. It's about how you interact with your environment. That's fitness. And they get that right.
Yeah.
Yeah.
It's so frustrating because it's so close.
We're almost here. I swear, everybody. I thank you all for bearing with me through all this stuff, but like we're doing it. I was back and forth all day. Do I do a short version or do we do the whole thing? And we just went with the whole thing. So here we are.
The kind of audience that we attract tend to like long form content.
So here we go. My dog cannot decide if he wants to be in this room or not in this room. That's why I keep disappearing and opening the door and then shutting the door. Okay. So what's going on here? Yes. This exactly. You can reduce it. Like reduction can be a selected for as a form of evolution. That's just that's a thing. Like we can observe that. And there's other contexts where it's not the case. Like if there's a food shortage, but there's another potential food, you could actually adapt to get a new biochemical trait to use that other food source.
But you lost a different function that is unrelated to that, so it doesn't count.
Yeah.
Apparently. That seems to be the conclusion.
But like this right here, the CYP, this is just a lie. The CYP mutation broke this regulation mechanism. It's not true. The original gene still exists. There was a gene duplication of CYP into another part of the genome. The original one was unaffected. You didn't lose anything. And to show how silly this argument is, you could just think about the opposite, right? Because what they're saying here is this CYT, what happened is, everybody, you have a gene that in order to do citrate metabolism, you need to express this gene. This gene, normally, it has a promoter. That's the on-off switch for the gene. Any biologists listening are like, just 101. It's the on-off switch for the gene. So you have this promoter, and that promoter is only active when there's no oxygen, right? Anaerobic. Now, this gene duplicates in this experiment in one of the 12 lines to another part of the genome. It doesn't take the promoter with it, just the gene duplicates. And it lands next to a promoter that's on under aerobic conditions. So now you can express that gene when there's oxygen, right? Now, the original version of that gene, unaffected. The new version, expressed in a different environment. That's what happened. Creationists say that is a loss of functionality because you have broken the regulatory mechanism for that gene. Putting aside, it's an on-ish office switch. That's a good way to describe promoters. I like that. That's the wrong comment. That's the comment. It's the on-ish office switch. Yeah, I like that for promoters. So let's pretend it was the opposite. Let's just say that E. coli had the ability to do both things ancestrally, right? You could do citrate aerobically, you could do it anaerobically. And then the gene for the aerobic that they needed got deleted. Do you think for a second creationists would say that was a gain of function?
No.
But that's what they're claiming must be the case if what we actually observed is a loss of function.
Yeah.
It's laughable.
Well,
it's not a serious argument.
That's one of the things is that for some reason, like, okay, creationists have this thing where apparently mutations always are a reduction in information. So it's like, okay, so if I have GATC and it mutates to GGTC, that's a loss of function. Okay, but what if the second G mutates to an A? I guess it's still a loss of function because it's a mutation. Okay, but you recovered the original genotype, so it should be an increase, right? Nope.
Yeah.
And there's no explanation as to why that's not the case.
That's interesting. I never thought of that.
They actually have this. The other one that I like is the loss of function versus a loss of specificity. So say you have an enzyme that has one target, right, at its active site. It takes a disaccharide, right, and breaks it into the monosaccharides. And then there's a mutation to that enzyme. And now it can target two similar disaccharides, right? Maybe it can target, maltose and sucrose, right? I don't know. I'm just making up, picking two disaccharides at random, right? Maybe it can target two of them instead of one of them. Creationists will say that if it's a loss of specificity, right? You've decreased your functional information right there. But what if you had an enzyme that could target two disaccharides and it mutated and now it can only target one? Loss of function. It did two things. Not only does one thing. Loss of function. Well, which is it? They can't both be a loss.
Dan, here's the crazy thing. Let's say we have a thing that can target one disaccharide, and then the gene is duplicated, and then the duplicate mutates to target two, and then it mutates to only target the one that the original gene didn't, which is a thing that happened. Apparently, the whole time, genetic information is going down, even now, though now you have two proteins with the same specificity targeting two different disaccharides, which is a gain in function versus the previous. It's still a loss in genetic information.
It's still, because each change they would call a loss. Yeah. Mm-hmm.
Yeah.
It makes no sense.
Because apparently, sure, that makes sense. Yeah. No, it doesn't.
I mean, the contortions here are funny, right? I mean, no new genetic information, novel gene function evolved. Like, this is just clearly not true, because aerobic citrate metabolism, and specifically aerobic CYT expression, is the new gene function that evolved. Like, it's like, this statement is just wrong. And you just need a citation for this. These beneficial mutations are only beneficial in the narrow sense, i.e. the actual definition of fitness.
Right, the one that we use in the discussion we're having.
In every case, they involve a loss of function. Really? Who checked that? I need a citation for that.
Well,
Yeah.
what they're going to say is just that the LTEE E. coli wouldn't fare as well in the wild as they do in the experiment.
Yeah.
And it's just like, yeah, I agree.
Yeah, it's context
because their
specific.
environment isn't the wild.
They cite that right here. Look, they say they have functions that are not used under the conditions. Yes.
Yeah,
It's
exactly.
context specific. Yes, exactly. But that doesn't matter. Okay. So now we're really at the end here. After they've butchered the LTEE. So I'm just going to bring this down here. Selection acts on the level of the whole organism. Selection does not happen at the nanoscale. Positive selection only sees those extremely rare, grossly beneficial mutations. This is not true. This gets the no.
Yeah.
The no is for this sentence. This is not true. The level of fitness effect that can be selected is due to population size and other things, but mostly population size. So that's just not true, right? It's context dependent.
I like how it's not just a no, it's a no. Like dot, dot, dot.
No, it's wrong. Why would you write that? Just no. That's what that is. Yeah.
This whole thing is it's so frustrating because at so many points you could have just not been dishonest, or you could have not pretended the definitions aren't what they were.
But no, you have to, because the whole argument hinges on that
right.
redefinition. That's the thing, and that's the, I don't want to call it a trick, but that's the trick. That's the bait and switch.
I mean, you're right. And it's frustrating because I want to attribute malintent to this paper for a number of reasons, but I'll just say this. This is to the authors. Y'all are making it hard.
I think that's fair. So now we're this is I just highlighted this because it's the big picture kind of summary thing, and then we get into the conclusion, and I just like fitness corresponsibility or probability of the organ. Yeah, like yes, that's what fitness is, which means it's not about complexity. It's not.
Mm-hmm.
It's yes. We show that Fisher's model is usually Fisher's model's famous theorem, so just they should be giving support to. Sorry, that sentence is weird. But basically shown that it's not right. They use how did you do this? Did they mention Mendel's accountant in here? Oh no, but they're like when realistic fact that's talked about like Mendel's accountant. Here we have this whole thing where they do the genetic meltdown, where they're unclear whether they're talking about the genetic the meltdown. Is that mutational meltdown or is it error catastrophe? We don't know. EV is extinction vortex. That's what that abbreviation is.
What is that gigantic initialism, though? FTNSWM? Like
fundamental theorem of natural selection with mutation.
No, stop that, guys. That's too many. that's too big of an initialism. This is a tongue-in-cheek criticism, but holy crap, guys! Like 5 should be your maximum letter number for initialisms.
Yes. So this is right. This is the idea right here. The skepticism regarding fitness is right. So fitness maximalization, right? They're skeptical that you're going to hit that ideal, right? You're not going to maximize your fitness.
Fair enough.
True. Fair enough. True. Here's the bait and switch. Mathematical models of natural selection provide no pathway to maximize fitness as net biological functionality. Here's the thing. The models are not dealing with functionality. The models, even Mendel's accountant, are dealing with the correct definition of fitness.
Mm-hmm.
Think about that. Everybody in the audience should think about that, because this is the central claim of this chapter. This line, this is the whole thing boiled down to one line. Right?
And the thing is, you think about it, if genetic entropy did lead to a reduction in fitness in the normal sense that evolutionary biology uses, it wouldn't be a problem because it wouldn't mean extinction. The creation need fitness from Mendel's accountant to mean reproductive success, because if your reproductive success doesn't decline, you're not going extinct.
Yep.
But they need these populations to go extinct, so they need their. Look, guys, you can't have your cake and eat it too.
this gets us to the end. This is it. So I want to end. We already talked about this. So I want to end with this, because this is the bait and switch of this paper, and I'm calling it a bait and switch, right? Remember, we start with Fisher is wrong.
Yep.
Here's the math why Fisher is wrong. Those models, including Mendel's accountant, are all based on the correct definition of fitness, reproductive success. Then they go on to say fitness should be defined as complexity rather than or functional, total functional content of the whatever the phrase is, right? It's about functional complexity, right?
They never
redefine.
So if you quote them poorly, I'm not going to super ding you on because they don't define it clearly.
Whatever the phrase is, it's about function, not reproduction, right? That's what they're saying should be the definition of fitness. And then they say, and look, in these experiments, we can document a decline in functionality in the genome, even as fitness increases.
Which
that
first
shows
can't.
Right.
They can't
but
actually
it shows, they claim, it shows that you can't maximize functional complexity. You can't, or more broadly, that you have a problem increasing functional complexity. That's the claim they're making here.
Which doesn't follow from any of this. It's
just wrong. It is just straight up a bait and switch. They're just changing they're making, it's a 3-part argument, and they're changing the definition of fitness partway through, and that's how they make the Fisher is wrong, therefore we have a fundamental problem for evolution argument. It requires changing fitness from reproduction to complexity. That's what they do here. They say it right here, as measured as net biological functionality. That's not what fitness is. And none of the mathematical models they talk about, from Fisher to Mendel's accountant, treat it that way.
I want to say a final thing to Sal. Sal, you came on my channel and gave an awesome discussion about why the second law of thermodynamics does not present a problem for evolution as a whole. It was awesome. And part of your discussion was talking about why common definitions for entropy by creationists don't actually have anything to do with thermodynamic properties, right? Where like chaos or disorder doesn't relate well to what entropy really means. That was a big part of your discussion. And I love that part of it because you were right. You were 100% right about that. What is happening in this paper is that you're doing the same thing to fitness. Those creationists you complained about, the entropy. They defined it in a way that has no relation to the actual field of science in discussion. Please don't make the same mistake in a different field that you complain about other creationists making in thermodynamics. Because you were so right about thermodynamics, but you're making the same mistake in evolutionary biology. Please don't do that. That's the last thing I wanted to Sal directly in this stream, because you were so right on thermodynamics and making that exact mistake here.
It's a good way to end it. Atheist Junior, do you have any parting words?
Sure. So I feel like this chapter, it's dressing up a lot of like lower tier creationist arguments, some really bad ones like quote mining Darwin about the eye and the ENCODE thing. So like on the surface, it seems like it's somewhat scientifically literate, but it's padded out with like a lot of math, a lot of repetition, but ultimately it's just got too many red flags, like too many misdefinitions, a bait and switch and mistakes. So to me, this is honestly just a glorified Answers in Genesis article. And I feel like I knew we were in trouble when I started reading the chapter and I saw the word evidences.
It is a red flag.
Yeah. And like talking about the LTEE, like they totally minimize the significance of like the citrate evolution. And I just think like if you're going to mention that, you should say how remarkable it is that Lenski has maintained that for 30 years, like going in on weekends, just the fact that it's gone on for so long, you know, I feel like you have to mention that. So that's all for me.
Yeah. Deadly Dakota Raptor.
Yeah, part of the things I find is the whole changing the fitness to the biological complexity and functionality is complicated designs are bad engineering. Complicated designs mess up, they interact poorly, they're not stable. The reason I define complicated code as spaghetti code, it's not good.
Yeah. That's and I want to for those of you that don't know, Deadly Dakota Raptor, our engineer who has not fallen victim to the Salem hypothesis. So I appreciate that.
I'll clap.
Thank you.
Applause, everybody.
Only one
nice to
round. No more.
My goodness. I don't know what it is with engineers.
I don't know. Yeah.
Actually, I do know. I do know. It's actually that engineers, the only way they get complicated processes done is by intelligent design, which fair enough. But then they import that idea to other aspects of the world where it's inappropriate.
All right, so let's do this part fast. Now, Deadly Dakota Raptor, correct me if I'm wrong, but you don't have like a channel, although you should.
Nope. Well,
no. Okay. Atheist Junior, what do you have coming up?
Right now I put out a video yesterday. Oh, and I put out my video about standing for truth, quietly editing the anti-vaxxer stuff out of his debate. And that video is my it's like, you know how YouTube says like your ranking by views of your last 10 videos. That's the #1 video for me. So it's got almost like 500 views in a day.
Classic.
You thought we wouldn't notice
propaganda.
You thought we wouldn't notice, but we did.
Oh, we noticed.
Yeah, and so I'm just I'm hoping that the video with me and Polochi will come out soon. I know he's got a lot of work to do on that video still, but yeah, so that's something to look forward to.
I can't wait.
Yeah,
Dapper, what do you have on the horizon?
So much. Okay, so tomorrow with Bent 70. That is going to be 6 p.m. Pacific, 9 p.m. Eastern. Thursday is going to be the next episode of Revenge of the Biddle, where I talk about Answers in Genesis stuff. And Biddle gave a speech about the 10 pillars of evolution, which, by the way, it was news to me that evolution had 10 particular pillars, but whatever.
Specifically 10. That's what they taught me in grad school, specifically 10.
This Saturday, there's nothing planned. So Tuesday the 12th is going to be a Kent with Bent pre-recorded because I'm going to be away. I'm going to be on vacation. If you try to contact me and you aren't basically like a personal friend, probably not going to respond.
Agree.
Yeah. Then Thursday, the last episode of Revenge of the Biddle should come out. Then that Saturday, again, it's probably nothing. Then the 19th, I'm going to actually see if I can set up like a Eric with Erica or a Matt with Maddie or even a Jackson with Jackson Wheat, talking about Dr. Jackson. But if not, it'll be a Kent with Bent. Thursday the 21st will be the release of one of my the versions where I take a previous series, it's broken 5 or 6 videos or whatever, and I turn it into one video. Then on the 23rd, I am currently, the plan is I'm going to be talking with Cindy Lincoln, Kent Hoven's ex, who he was just convicted of abusing and sending to the hospital, and he now has to pay her medical bills. He's going to be going to prison or jail, for 30 days. He's going to be on a 1-year patient on this whole thing there. Then on the 26th, it'll be more. Or sorry, the 25th should be a debate between Dr. Dan here and Finding Truth, a Muslim apologist who is an engineer. The 26th will be another Kent with Bent. Then on the 28th, I should start.
How do you keep up with this schedule?
I do YouTube stuff like 12 hours plus a day, man.
What's on the 28th?
The 28th should be the first episode. Unless it's a speculative evolution video, which might happen, the 28th should be the first episode where I take a look at Cindy Lincoln's tour of Dinosaur Adventureland Science Center and explain to her specifically why, no, this is not good evidence for Kent Hoven's views.
So that's a separate video with her? Is there 2 videos?
So it's not so much with her in the sense that she was present for this. So basically, Cindy Lincoln on Facebook said, okay, I know that Kent Hoven is a lying, abusing jerk. But what do you have to say about his amazing evidence? And this is my response to this. Like, well, let's take a look at his amazing evidence as you yourself have presented it on his property. And so in that, I'm normally a little bit snarky and a little memey, but in this series, I'm very nice and very understanding, and I'm pointing out all the time where I think that it's not Cindy who's getting it wrong, it's Kent who's speaking through her.
Right.
So, yeah. Oh, and there's
Hi, dogs. Thank you, Dapper. You keep up an impressive schedule and just pump out content. I don't know how you do it.
Oh, I do 12 plus hours a day.
Yeah.
If I could just say something about Cindy Lincoln real quick.
Please go ahead.
Yeah, obviously I did an interview with her before, and I will say, obviously, like, when it comes to, maybe theological or scientific beliefs, obviously we have some major disagreements with her, but I think ultimately what's important is people's safety. And so
Agreed.
I think that's the one thing to remember. And, I think, we can talk about these things without having to be, snarky or whatever. Like, there's certainly some creationists who deserve your ire, like Kent Hovind, but I would say that she and Sal are not one of them. So,
yeah, there's a reason that I'm not directing my audience to her versions of these videos, because I'm not linking the original. Like, I expect most of my audience to be nice and awesome, but there's always the outliers, and it's just like, this woman does not deserve any harassment or anything like that. And that's a great advice. Get vaccinated.
yes.
I'm waiting for my approval for a booster shot, because I currently don't have it, but
You should.
should I?
I think, well, we'll talk off the air, but yes, you should get a booster shot.
Well,
once you're 6 months past your second shot.
Don't think I am.
Well, that's that problem. We'll talk later. So what I got coming down the pike is next Monday, 1 week from today, I'm going to talk with David Neff. We're going to have him on because he went to an event with Dr. Rob Carter. There's like 3 hours of video from it. So I am going to I'm going to watch it because it's available online. So be on the lookout for that one. And then on Wednesday of next week, I'm going to appear on Sal Cordova's channel, Evidence and Reasons, to talk about this chapter. We're going to talk about it. I'm really excited for that conversation. Sal and I always seem to have really interesting conversations about this kind of stuff.
I'm a little
so
jealous.
I'm really looking forward to that. I mean, you should ask Sal, see if you can come. He's cool.
Well, I'll be away.
Oh, that's right. You'll be all right. You won't be around.
Yeah.
All right. So that's all I have directly coming up. I get those two things. So that's what I'm doing soon. And I hope to see you all in those two places. So my goodness, we've been going for 2.5 hours, but we wanted to be comprehensive, and I feel like we were.
Yeah.
So
happy
thank
4th
you
day
all
of
for
Halloween.
watching. Oh, what was that?
I said happy 4th day of Halloween.
All right, everybody. So thank you for watching. We're gonna call it a night. I'm tired. It's late. So I can't believe we did that whole thing. But thank you, everybody. I appreciate all of you for watching. Have a great night. Like, subscribe, share, do all the stuff. Do it for me. Do it for Dapper. Do it for Atheist Jr. Do it for
Turn on notifications.
Turn on notifications. All right. I'm not going to keep this going any longer. Good night, everybody.